Good afternoon, everyone. I'm delighted to welcome all of our viewers to the VTC School of Medicine Research Symposium for the class of 2020. Now, it may sound like more than 10 people are in this room, and don't make me turn the cameras around, but I'm counting, and we're good, okay? So I've been looking forward to this event since becoming dean last July, and back then I never could have imagined that we would be streaming this to you in a large auditorium occupied by less than 10 individuals observing social distancing, and that we would be featuring virtual instead of live poster presentations. The symposium will be a welcomed break for our understandable preoccupation with the COVID-19 pandemic. Research is not just one of our 4 curricular domains, it is one of the defining strengths of our medical school as we pursue our mission of creating future thought leaders in medicine. These thought leaders are scientist physicians, they are inquisitive and apply the rigors of scientific thought to clinical practice. This is the VTC difference that distinguishes our graduates when they start their residencies. Congratulations are in order for each member of the class of 2020 for fulfilling the rigorous requirements of our research domain, along with heartfelt thanks for all of the support and encouragement provided by your faculty mentors. It is now my pleasure to introduce Dr. Mike Friedlander, a man of many hats at Virginia Tech and the Fralin Biomedical Research Institute. Mike is here today as Senior Dean for Research in the VTC School of Medicine. I greatly appreciate the leadership provided by both him and Dr. Leslie LeConte in assuring an exceptional research experience for our students. Mike?
Thank you very much, Lee. It is certainly a pleasure to be here. I was thinking back to a little over 10 years ago when we turned in a report for the AAMC called The Scientific Foundations of Future Physicians. Little did I understand at the time or appreciate what that would mean in terms of what's happened at this incredible School of Medicine. And you, the medical students graduating this year, the faculty, all of you, are really part of that, the living embodiment of what was meant by that very important report for the future of American medicine. All of you go forth soon, graduating, and you will be on the front lines of what you can see right now is one of the most challenging periods in history in terms of the delivery of health care. And you will be taking with you the skills and clinical practice you've picked up here, the scientific foundations, and doing the great service that we all need. And I can't tell you how much I appreciate all of you for doing that. Another part of what you will carry forward is what you've learned and been involved with in terms of research. And I always like to say, for all the families and the people you will touch in the clinic, and there will be many, and the difference you will make, and it will be large, there's another component through the research that you have done, are doing, and will continue to do that will touch the lives of so many, those you don't even see living today and those yet to come. So the research mission may in some ways to many on the outside right now seem a little removed from the immediate needs of delivering clinical care, but I assure you what you have done, your excellence in research, and the knowledge you have obtained here at Virginia Tech Carilion School of Medicine will have an impact on the world for literally generations to come. So it is really my great pleasure to be part of this day with you and to share in the experiences of today. Now, as part of the program, I have the great honor at this point to say a little bit about recognizing the faculty mentors. For all of you students out there, you know the mentorship you've received from folks in Carilion Clinic, at the FBRI and Blacksburg campus. These are faculty that are passionate and are dedicated to working with you and helping in your research. And you've all done fantastic jobs with them. But we want to take a moment to recognize those faculty mentors who contribute so much to the overall process here at the School of Medicine. And in particular, as you know, the environment here that we've tried to create is one that enables you to bring forth your own ideas, come up with a hypothesis-driven research project that's original, and come up with opportunities for your own professional growth. And also, we'd like to recognize mentors who embody these things and also challenge their mentees to make original contributions, foster your intellectual growth, not just come into my clinic or a lab and do this. Here's some data to analyze. Here's some reagent to put in a test tube and analyze it and so forth. As you know, you do so much more than that. But to allow you to use your brilliance, your knowledge, and your passion to really make a difference on your project. And also, these mentors have to establish clear guidelines and goals working with you so that the project really comes to fruition. There are so many that we could recognize, so many that have done so much, but the contributions to being a mentor here at the School of Medicine. And this year's awardee is Dr. Dev, who many of you know, who has been an outstanding exemplar of what we look for in the mentors. She is currently mentoring four medical students to show you something about her commitment to this mission and how important it is to the School of Medicine.
What particular areas I had a personal interest in, and she helped me develop my project out of that.
She really gives me that creative space to design my own project and come up with my own research question. And I feel like in the research world, it's so important for me to have that experience to grow by myself, but at the same time, I know that I have her as a resource.
The best way to help them learn is really to have them put their hands in and participate. To also be able to generate their own ideas and to make it their own. Because I think when you're passionate about learning about something, you want to feel that you're part and parcel in moving that for that field forward in some ways.
She's always got like high spirits, which is very encouraging when you're dealing with a difficult research project that sometimes you can get kind of down in the dumps about if things aren't moving quickly enough or there's so much paperwork in the way. And so she's always kind of very supportive in that regard.
I think that in this profession, from what I've seen, it's really easy for people to kind of get caught up in day-to-day activities and not focus on why they're doing what they're doing. But she loves her patient, she loves her projects, and she puts her passion into everything.
I think it has a huge impact on how your research goes, is how involved your research mentor is and how responsive they are, and she's always been fantastic in that regard. I consider myself very lucky and fortunate to have this type of mentor and relationship in my career.
You can just tell that she's so passionate about her work and her research, and I really hope that I'm able to emulate some of these qualities that I've just seen in her.
I feel that part of me is just so excited about what I do. And I feel if somebody is as excited or becomes that excited, then I think we're in harmony and we can all produce together. So it's so great that they feel the passion that I do. I can't tell you how amazing it is to be thought of and to be considered for this award. It makes me feel so good inside.
Please join me in welcoming Dr. Dev to the stage to receive her award. Dr. Dev.
Thank the students, the selection committee, and all of the mentors and other students that are here for considering me for this, this wonderful award. I am truly honored and truly grateful for this award. And I'd like to thank particularly my students who are, as Dr. Friedlander mentioned, really very smart and thoughtful and have the passion and to move, as I say, the needle forward in our interests and what we do. I also want to thank the mentors who have taught us and on whose, from whom we've learned and who have laid the foundations for us to learn from and have moved science forward. And as a mentor, and one that has also had mentors, we stand on their shoulders and we are thankful to them. And many of the students also will have not just one mentor, but many mentors. And our goal in mentoring is hopefully to have them stand at the very top while we shoulder them. And so I'm really excited that I've had the honor and the opportunity to work with some amazing students. And I'd like to go on and highlight what some of these things, why I think they've done such an amazing job. One of the things about research, I think, is that we think of the solution there, we just have to search it again. So in research, I think the goal is to be able to look for the solution and to be able to not just look for it, but look for it in a rigorous way so that we can find what the real answer is, and to be able to repeat that and make sure that if we find a solution, then that is truly what it is. So whoever repeats it in the same way that we look at it, it should bring on the same answers. The population, my population that we are looking at are the chronic kidney disease population. And so the projects that we are looking at, or the students are looking at, come out of the population that they have studied. And you can see that we start with not just patients that have chronic kidney disease or acute kidney disease or renal transplant or requiring therapy, but all those that also start at risk. So the risks that get them to that position. So as we move forward, we know that this is a disease that encompasses many, many people, and as a result, comes with many risk factors. So in highlighting how we can understand that, we have a project that looks at the various risk factors and how to impact those risk factors in relation to not just the population we have, but in a broader sense if feasible. So we have a project that the students have participated in that's a long-going pragmatic project, but out of that comes many other projects. And that we call that the Program to Increase Chronic Kidney Disease Education, Test Diet and Health Outcomes. And so the acronym is picked out. We have designed it as a long-term project that is involved both survey and a randomization of patients. And the overall arching hypothesis looks at not just kidney education at risk patients, but also in their caregivers and how we become aware of the disease, how we enforce various lifestyles and improve parameters that lead to kidney disease and to preserve kidney function in the long run. And to that end, with the larger hypothesis, there are multiple aims to this study. And having the opportunity to look at various aspects, it was really heartwarming to know that when this was presented at Research Live, that Manavi Bhagwat, who is one of the third-year students at the present time, took immediate interest in looking at what things might happen if we were able to change diet. And so her project actually took on her role where we looked at common diets that we prescribe to prevent kidney disease, and if we extend it to risk factors, which are now known for improving that, how that might impact how it was perceived both at the patient's level and how the outcomes might vary based on what the patient did. So she took on that project and has been really wonderful in progressing and in doing her randomizations and looking at her patients and recruiting them actively. She's passionate about that, and as we are about answers and thinking about it. So it's been a really wonderful a wonderful relationship to know that this project is hers and she's taken it moving forward in 100 ways. The other projects that may stem from this also were passionate about that Varun took on, Varun Carver, also a third-year student at this time, and he wanted to see how we could actually make a difference in looking at early diagnosis. And with some of the biomedical engineering mentors at Virginia Tech, took on a project which looked at how a molecular spectroscopic urine analysis can look at early kidney disease. In that end, he not just looked at how to possibly determine kidney disease, but early on, but how to look at or possibly evaluate for glomerular disease, particularly diabetic kidney disease. And I want to mention that going back to Manavi's project, because of that project, certain questions came up on their own as to, what do we know not just about the patient, but how do we treat, how to mentor, physicians actually take on what we need to do to teach our patients appropriately. And so when that came on and there was not a lot of information, she immediately took to her project, created an amazing survey, put it together as efficiently as possible with literature backup, and she was able to present this at the American Society of Nephrology. And I have to tell you, in 2018, and she did this with such passion and wonder that she actually had the Helio social media come and seek her out and give her an interview. So you can see that she really took to it and has obviously enjoying her presentation. And she got lots of people come and visit and give her wonderful information and camaraderie, which is really helpful when students go to present. In addition, she took further on to understand about excretion of various solutes in again learning about her project. And we came upon an interesting patient presentation, which she thought she would look much deeper into, and then actually wrote about this and as an abstract, and she got accepted for the American College of Physicians Virginia chapter oral presentations for her. So one project leads to another, and I have to tell you that it's the students' aspect that moves us forward. On Varun's aspect, I'm really proud to say that as for these last few days, we've had the National Kidney Foundation meetings, and he, even though it's become a virtual meeting at this point, he has the opportunity to have this abstract accepted. This is part of his project where he took on the spectral characteristics of chronic kidney disease and is virtually presenting it at the conference. Again, you can see that a lot of effort went into it. He had quite a few mentors, and he took the interest of learning about this technique specifically and knowing it in such a way that he can actually present and give us a very good understanding. And he's made a difference in that we are seeing early results showing that there is some separation and perhaps might be a tool in diagnosing kidney disease in an earlier form. Furthermore, he also took on interest in presenting and having a nice case which looked at calciphylaxis, unusual case in an unusual presentation, but the interest to be looking into this, to write it up, was Varun's, and he also got this accepted as a presentation at the National Kidney Foundation, which he will be presenting or has presented as in a virtual sense. And out of them comes various articles. I have to mention Manavi, because of her abstract presentation, she has already written up a manuscript, and we are submitting that. In addition, Varun has, with his mentors, has published not just research articles, as you can see, several of them participating in the group, but also his own review article, which you can see comes as a result of their interest and the result of moving forward the needle as we speak with the passion that they have. I want to also mention that student second year, Hiba Zafar, also took on interest as we presented in Research Live, which is a really nice forum for the mentors to bring projects that need a voice, that they can give their passion. And it's lovely when the students comes to you and says, I really think this is really interesting, and then they can develop from that. So we were interested in looking at the chronobiology of hypertension, particularly in the night, in relation to mineralocorticoid receptor antagonists. So Hiba took on this project, which she developed, again, the population she was going to study, how it was going to proceed, what she wanted to do, how to randomize. So I think it's really very important that she gave life to this very interesting project, and we are looking for upcoming results in the near future. And last but not least, Quinn Weinberg, also a current second-year student, took on the project where she wanted to look at spectral urine spectroscopy again, but not in the sense of specific chronic kidney disease, but in the sense of looking at renal cancers. And how, if we could make a difference in early diagnosis, because there may be a role. And in her project, because there had been some early data suggesting that they may be able to trace cancer in bladder, we thought she chose to look at it in the kidneys. So again, a project that's exciting, a project that's enrolling, a project that she's passionate about, and I am equally passionate with her, and I look forward to her results. And last but not least, I'll end. And here I want to say thank you, and I want to say that this award is not just for me, but the award I accept on behalf of all the mentors that are working so hard, and all the students that really are juggling a lot of work that is medical school, which by no means is small rigor. On top of that, taking passionate interest and learning, and not just that, but bringing their thoughts and their ideas, which I think always makes research even better when someone else can give their ideas and thoughts to the process. So I accept the award on behalf of all of the mentors and all of the students, and thank you all.
Thank you everyone for being here, the several people in this room. My name is Anisha Chadha. My project is entitled Vascular Remodeling in Preeclampsia, Smooth Muscle and Collagen Content in Placental Vessels, and my mentors are Dr. John Chappell and Dr. Allison Dorica. I have no conflicts of interest to disclose. So preeclampsia is a serious pregnancy complication that affects 2 to 5% of pregnancies worldwide. In fact, it is one of the most common causes of maternal morbidity and mortality worldwide. Currently, the only known and successful treatment of preeclampsia is actually delivery, and specifically, it's delivery of the placenta. Symptoms typically resolve in preeclampsia between 48 and 72 hours. Thus, it is the placenta, not the fetus, that is thought to be the main culprit behind the pathogenesis of preeclampsia. So though the molecular pathogenesis of preeclampsia is still incompletely understood, it is thought to occur in part due to the abnormal vascularization of the placenta. So the placenta is highly organized in order to facilitate optimal fetal and maternal oxygen exchange. Zooming in here, we could see that the maternal spiral arteries feed the lacunae surrounding the functional units of the placenta, which are called the terminal units, where fetal capillaries thereby take up oxygen and nutrients to nourish the developing fetus. So these are the most important structures for oxygen and nutrient exchange, thus these are the areas that we were interested in exploring. So next I'll detail what is known about aberrant remodeling of the spiral arteries that is thought to be central to the pathogenesis of preeclampsia. So early in gestation, fetal cytotrophoblasts invade the myometrium surrounding uterine spiral arteries. This prompts the spiral arteries to become low resistance, high capacitance vessels. Thus, this is capable of adequately perfusing the placenta and nourishing the developing fetus. In preeclampsia, however, cytotrophoblast invasion is limited to the decidua and does not invade the myometrium. Thus, the spiral arteries remain high-resistance, low-capacitance vessels. This causes inadequate placental perfusion and placental hypoxia, so the fetus is not able to get enough oxygen. So the ability of the cytotrophoblast to invade the myometrial spiral arteries really is influenced by a balance of pro- and anti-angiogenic factors. So a healthy endothelium can be maintained when the levels of pro-angiogenic factors are high, including placental growth factor, or PLGF, and vascular endothelial growth factor, or VEGF, and when the levels of anti-angiogenic factors are low, namely sFlt1, or soluble FMS-like tyrosine kinase 1. So the main source of sFlt1 during pregnancy is the placental syncytial trophoblasts. In preeclampsia, sFlt1 has been shown to be overexpressed. This causes an inhibition of both PLGF and VEGF, as well as an inhibition of cytotrophoblast invasion in vitro, thus causing overall endothelial dysfunction. In fact, the in vivo removal of sFlt1 during pregnancy in severely preeclamptic women has actually been shown to decrease the symptoms of preeclampsia. So the gap in knowledge that our lab was interested in exploring is how fetal vessels remodel in the functional unit to compensate for aberrant spiral remodeling and sFlt1 overexpression in this chronically hypoxic preeclamptic placenta. We utilized 2 organ systems in order to build our hypothesis. One, we use the lungs. So in pulmonary arterial hypertension, chronic hypoxia causes normally non-muscular arteries to become heavily muscularized. You can see on the left here that the vessels are very thin in order to allow for diffusion of oxygen. But in pulmonary arterioles in PAH, they're very thick. So you can imagine that these vessels would have a hard time diffusing oxygen. In systemic hypertension, cardiomyocytes, or cardiac tissue, has been shown to increase type 3 collagen content to overcome for the upstream resistance generated by systemic vessels. Type 3 collagen is a component of reticular fibrils that is found in vessel walls and adds structural support. Furthermore, our lab was also interested in the role that pericytes may play in this remodeling process. Pericytes are a cell type that surround endothelial cells and play a crucial role in maintaining microvessel stability and tone. Pericytes are thought to be located in a unique place in that terminal unit, and their close link to endothelial cells may afford them the ability to sense the blood in the intervillous spaces and sense if there's placental hypoxia. Studies have shown that pericytes can actually differentiate into contractile smooth muscle cells under chronic hypoxic conditions. So it goes to show that in the chronic hypoxic condition of preeclampsia, pericytes may play a key role in muscularizing the vasculature. To the best of our knowledge, there's actually no data on pericyte coverage in placental beds, despite their demonstrated importance in other vascular networks, most notably the brain. Our lab has preliminary data that shows pericyte coverage in a murine placenta at day 14.5 in a mouse placenta. And this corresponds to about a second trimester placenta in humans. So to summarize, our hypothesis is that there will be increased smooth muscle and type 3 collagen content around placental fetal vessels in preeclampsia, and there will be increased pericyte coverage surrounding the vessel walls of preeclamptic placentas. For our methodology, first we identified and consented eligible subjects at Carillion Roanoke Memorial Hospital. In the end, we identified 42 subjects. These subjects had a diagnosis of preeclampsia with or without Savitcher's or HELP syndrome. We also identified normotensive controls that were gestational age matched. We excluded those patients who had a previous diagnosis of chronic hypertension, multiple gestations, or gestational hypertension. We performed chart review in order to obtain the estimated age, gestational age at delivery, the type of delivery, and the patient's smoking history. Next, we took placental cores in each of the 4 quadrants of the placenta and then processed the tissue. For the pilot phase of the study, the clinical team at CRMH actually chose 10 subjects. 5 subjects had a diagnosis of preeclampsia, and 5 were normotensive control subjects. The research team was blinded to the diagnosis. We performed sectioning of each placental core, and then performed immunostaining in order to identify salient features. We identified endothelial cells, smooth muscle cells, collagen type 3, pericytes, and cell nuclei. Lastly, we performed imaging with confocal microscopy to identify representative villi at 40x magnification. We characterized the vessel morphology and also performed Z-stacking in order to view the vessels in a Z dimension. So for our results, these are the total subjects that we enrolled in the study by the diagnosis. We enrolled 20 normotensive subjects and 22 subjects that had some form of preeclampsia. For our pilot phase, these are the subjects that we enrolled. We had 2 who had preeclampsia with severe features, 2 had preeclampsia without severe features, and 1 who actually had postpartum preeclampsia, which is a very rare condition where a patient is born, delivers who is normotensive, but then subsequently develops the features and symptoms of preeclampsia, and this patient specifically developed them about 3 to 4 days after delivery. So the images I have here depict representative confocal images that I obtained for these subjects. So I included these images on the left, which is the images I obtained, as well as these images on the right to depict the features that we were trying to analyze. So to characterize smooth muscle content, I measured the area of smooth muscle actin positivity surrounding each vessel and compared the 2 groups using a mixed model statistical analysis. As you can see, the vessels in the normotensive placentas had a significantly thicker layer of smooth muscle relative to the vessels surrounding the normotensive placentas. The mixed model analysis also confirmed this. Compared to the vessels within the placentas of normotensive patients, vessels from placentas of preeclamptic patients demonstrated a significantly reduced area of alpha smooth muscle actin positivity, 2.48 to 3.97, with a p-value of 0.0076. The total percentage of smooth muscle actin positivity across the villus surface was not significantly different, and the proportion of vessels that were alpha smooth muscle actin positive were not significantly different in either group. I utilized the same methodology to analyze and quantify the collagen III content. Despite the increased vascular resistance that we see in the placental vascular circuit of a preeclamptic patient, we actually again see less collagen III in the preeclamptic placental vessels than the normotensive vessels. This is also reflected again in the mixed model analysis, so a trend was seen towards decreased collagen content surrounding the vessels and the preeclamptic vessels. Again, the collagen III positivity was not significantly different in either group, although you can see that the amount of collagen III positivity was decreased in the preeclamptic vessels. And the chi-squared analysis showed that the proportion of collagen III positive vessels was not significantly different. So to summarize our findings, contrary to our hypothesis, the vessels from placentas of preeclamptic patients demonstrated a significantly reduced perivascular smooth muscle content. Despite the increased resistance, upstream resistance like we talked about with the aberrant spiral remodeling in the maternal vascular circuit, the fetal vessels and villi in the preeclamptic placentas had decreased type 3 collagen content. Finally, though causation cannot be inferred from this cross-sectional study, we theorize that placental vessels may in fact compensate for the decreased perfusion by decreasing vessel wall muscularization and collagenization, thus facilitating increased transplacental oxygen exchange. So this study overall gives us a better understanding of the potential downstream effects of aberrant spiral artery remodeling, and it could lend and posit how this might also be related to sFlt1 overexpression. For example, if sFlt1 is overexpressed and inhibits proangiogenic factors such as VEGF and PLGF, it could in fact have played a role in limiting smooth muscle proliferation and content in these fetal vessels. Furthermore, preeclampsia is an important cause of iatrogenic prematurity. So better histological classification of preeclamptic placentas postpartum could in fact identify at-risk women in their future pregnancies. We know that with preeclampsia in one pregnancy, women are at a very increased risk of developing preeclampsia in subsequent pregnancies. So by identifying these women, we could deliver earlier intervention and more aggressive prenatal monitoring. Lastly, to shift gears and again talk about pericytes, we did find evidence of mural cells positive for NG2, which is the pericyte marker, within the vessel walls of the preeclamptic placenta, within both placental, The preliminary mixed model analysis revealed that there was no significant difference between the proportion of pericytes surrounding the capillaries of the normotensive versus the preeclamptic placental terminal villi. So further studies should be undertaken to reveal whether, if at all, these pericytes actually play a role in the demonstrated vascular remodeling. So while we analyzed over 342 vessels from each of the 10 placentas included in the pilot phase of the study, these findings should be replicated in the remainder of our available placentas. So I'd like to emphasize that this is an ongoing analysis. In order to overcome patient-to-patient, sample-to-sample variability, we would need 28 total placentas, 14 normotensive and 14 preeclamptic. We would also like to investigate how maternal sFlt1 levels are correlated with the demonstrated differences in smooth muscle and collagen expression to add to the growing body of knowledge that supports the importance of sFlt1 in the pathogenesis of preeclampsia. Lastly, we'd like to include those with a diagnosis of either chronic hypertension or chronic hypertension with superimposed preeclampsia. These patients represent a really important subsegment of the patient population, and this would allow for a more comprehensive evaluation of a specific type of placental hypoxia and possibly decreased placental perfusion. I'd like to acknowledge the members of the Chapel Laboratory, including Dr. John Chappell, Sarah Taylor, our lab manager, Dr. Laura Beth Payne, Malek Boozaher, Stephanie Masters, and Justin Davis. I would like to especially acknowledge the patients who donated tissue for this study, the CRMH OBGYN department, including Dr. Allison Dorica, Dr. Megan Lord, and Dr. Ha Nguyen. And I'd especially like to thank Dr. LeConte and Dr. Allison Teggy for statistical support. Thank you, and I'm open for questions now.
Okay, so while I wait for the YouTube questions to come in, we have a few here in the audience. So, Mike,
So,
Dr. Friedlander.
First of all, that was outstanding project, outstanding presentation, sincerely. You brought up something that got me thinking about the compensatory changes, and that is about the dynamics. And I realize you're not able to go into different time points and
Yes.
longitudinally sample, but I'm just asking you to speculate a little bit in terms of the findings that you got. They were a little surprising, I would say, very interesting. Can you speculate a role of what some of the early phases might have been different and what you're seeing is a later compensation possibly, or what do you think?
In terms of whether we were able to analyze placentas at an earlier gestational age versus a later.
You could longitudinally sample one at different times.
That's actually a really interesting question, is one that I was actually interested in exploring. So unfortunately with gestational age, and when you're trying to evaluate differences in placentas at different gestational ages, it's kind of hard because being premature and having premature placentas in general, it could be that these placentas have pathologies not actually just related to preeclampsia. So I think it does add a lot of different variables that we can't really account for, but it would be very interesting to see how, whether these vessels are unstable or like have different architecture than the later vessels, for sure. Dr. Lerman?
Yes, I'm wearing my OBGYN hat at the moment. Thank you. That was a wonderful presentation, and I'm so glad we're looking into the mystery of the placenta and an underappreciated organ, if you ask any pathologist around. In preeclampsia, we're talking about a syndrome of findings that have yet to have a clear cause. And as you build the case for this potentially being in the pathophysiologic lineup, have you considered doing any dose-response types of studies? I know it will take a larger study to find those severe preeclamptics. You mentioned the chronic hypertensives, and that's a good population also. But amongst the preeclamptics, if there was a way to kind of grade the phenomenon in severity, either using the clinical criteria or other variables, it might help you to build a case for cause and effect here.
Yes, definitely. So as I mentioned, we have 42 patients, and they're across the preeclampsia is really a spectrum. There's patients without severe features, so they don't have end organ damage, meaning this is for not Dr. Lerman, this is for everyone else in the room, but they might have headaches, they might have liver damage, they have a host of other features that really make delivery more imminent and make patients more sick. So what Dr. Lerman is saying is definitely true. So I think with ongoing analysis, we'd definitely be able to grade these placentas by severity. And with our initial analysis, something that I haven't shown on here quite yet is that we do see a dose response, but I think with 42 placentas, we'd actually be able to really characterize it and make a case for it.
Okay, we're going to take one quick question from YouTube. A couple of good ones came in. So David O'Neill asks, you mentioned decreased collagen possibly being compensatory, but could it also be a result of global ischemia preventing appropriate growth or energy to develop collagen?
That is definitely true. Again, causation can't be inferred from a cross-sectional study, so I can't say for certain whether this is a cause or effect, but I do want to emphasize that what we know happens in preeclampsia is S-split overexpression, so I really think that these are downstream effects from aberrant by remodeling and S-split overexpression.
Excellent. Thank you so much, Anisha. Our next speaker is going to be Chris Lau, and he worked with Ian Kimbrough and Dr. Harry Sondheimer at FBRI. And so we're going to go ahead and get him set up to hear a little bit about his research.
We're ready. Hey everyone, thanks for being here, and for all those on Zoom, thanks for listening in. My name is Christopher Liao, and today I have the privilege of sharing my work on the effects of amyloid beta on the astrocyte vascular interface. And first of all, I just want to take this opportunity to thank everybody who's helped me get to this point and allowing me to present the project I've started 3.5 years ago when I first arrived at VTC. So to start off, I just wanted to make sure we're on the same page. I wanted to start with some background just to help bring my research into context. Cerebral amyloid angiopathy is a condition caused by the accumulation of vascular amyloid around blood vessels in the brain and can lead to a variety of clinical deficits and conditions. This is a histological cross-section of a blood vessel in the brain. And here there's vascular amyloid actually stained in brown. And everyone, thanks for being here, and for all those on Zoom, thanks for listening in. My name is Christopher Liao, and today I have the privilege of sharing my work on the effects of amyloid beta on the astrocyte vascular interface. And first of all, I just want to take this opportunity to thank everybody who's helped me get to this point and allowing me to present the project I've started 3.5 years ago when I first arrived at VTC. So to start off, I just wanted to make sure we're on the same page. I wanted to start with some background just to help bring my research into context. Cerebral amyloid angiopathy is a condition caused by the accumulation of vascular amyloid around blood vessels in the brain and can lead to a variety of clinical deficits and conditions. This is a histological cross-section of a blood vessel in the brain. And here there's vascular amyloid actually stained in brown, and you can see that encasing the blood vessel circumferentially here. And when this problem becomes diffuse and widespread, it can cause a variety of pathologic bleeding events in the brain that could lead to clinical neurological deficits, and it can also lead to deranged cerebral perfusion. So because of this, CAA has been associated and linked to patients suffering from dementia and patients with Alzheimer's disease. But unfortunately, current treatment options are limited, and the pathogenesis is not well understood. So we decided to study astrocytes because although CAA, dementia, and Alzheimer's disease have been studied in a neuronal context, glial pathology has not been as thoroughly investigated. Furthermore, recent research shows that there's evidence to suggest that patients with Alzheimer's disease have been associated with reduced cerebral blood flow. And you can see here that cerebral blood flow relies significantly on a very complex interaction between the astrocytes and the blood vessels. And in fact, the astrocytes are responsible for much of the maintenance of the blood-brain barrier integrity. Importantly, the astrocytes actually communicate with the blood vessel cells through their long and numerous projections that terminate as astrocytic end feet. And these end feet actually make direct contact with the blood vessal wall in their cells and communicate biochemically with it. And soon you'll see that these astrocytic end feet are actually key elements of our research. In fact, what initially inspired this project was that my mentor published a paper showing that in aged mice, vascular amyloid actually forms a rigid cast with their plaque deposits around cerebral blood vessels. And here's a three-dimensional reconstruction of that. Basically, the blood vessels are shown in magenta, and the vascular amyloid plaques are shown in green. The authors also showed that in control mice, the astrocytes on an electron microscope actually, their end feet covered circumferentially and directly the endothelial cells here labeled in purple. Whereas in mutant mice, actually, there were significant amounts of vascular amyloid that ended up occupying the space between these cells, thereby impairing the ability of the astrocytes to regulate blood flow to the brain. Additionally, further research in human tissue has uncovered similar findings and sort of reinforced the clinical relevance of what was published before. And basically what this graph shows is that in cognitively intact, healthy patients, there is good astrocytic end-foot opposition with the cerebral blood vasculature. But in patients with Alzheimer's disease, the astrocytic end feet are displaced, and there's a reduced amount of contact between the end foot and the blood vessels. So currently what we don't know is how this process of displacement of the astrocytic end feet from the blood vessel actually happens, and whether it precedes or follows overt amyloid plaque deposition. Here's a diagram illustrating the process of amyloidosis, and it's important for us to understand that amyloid beta actually exists on a continuum of species, beginning first as a monomer, and then progressively aggregating into low molecular weight oligomers, diffusible protofibrils, insoluble fibrils, and then finally the rigid plaques that we talked about before. We wanted to focus on the low molecular weight oligomers and the soluble forms of amyloid beta because evidence suggests that these soluble forms can actually act as a ligand and bind to specific receptors in the brain. And if these oligomers and ligands accumulate to toxic levels, they can actually start to cause aberrations in cellular function. We also believe that MMPs, matrix metalloproteases, were somehow involved in this process. MMPs, matrix metalloproteases, are digestive enzymes that are normally helpful for the body. They function to clear foreign proteins from the body, such as amyloid beta, but if they accumulate to excessive levels, they can start to degrade everything else in their path. So tying this all together, we formulated our primary research question, which was how do soluble amyloid beta oligomers contribute to disruption of the astrocyte vascular interface? We hypothesized that the oligomers do this by promoting MMP release, which causes the degradation of the basement membrane between the astrocytic end feet and the endothelial cells in the blood vessel. To start tackling these questions, we started by validating our preparation of these oligomers in the lab using a Western blot. We injected these oligomers into animals, into mice, by mirroring a protocol that was published in 2016. And in this paper, they basically used the same exact protocol and found behavioral deficits in spatial memory. We then fixed and sliced the brains for processing in preparation for immunofluorescence staining and quantification using various software. Here are the results that we found. So on the left here is a Western blot showing that we were able to actually synthesize the oligomers in the lab, starting from a monomeric stock powder. Here's a schematic of the injection protocol we used. In total, took 2 weeks, and then we injected the mice. We also injected them with dye. And here, this image basically shows that successful penetration into the intraventricular space. Here are some microscope images showing that after 2 weeks, compared to the control animals, the experimental animals actually had an elevated, significantly higher amounts of soluble oligomeric forms of amyloid beta in the absence of insoluble fibrils that were labeled by LOC antibody and rigid plaques labeled by methoxyXO4. Furthermore, we found that there was significantly upregulated reactive microgliosis and astrogliosis here in the brain, particularly corresponding to areas that had a high amount of these amyloid beta oligomers. And in these heat images, basically black represents zero intensity of the immunofluorescence stain, and then yellow represents maximum intensity. When we use a higher magnification view, we can see these astrocytes and microglia in more detail. And fortunately, this higher magnification allows us to see that the microglia actually exhibit not only increased overall expression, but also altered morphology as well. When we decided to look more closely, we found even more supporting evidence that the microglia may have an even more important role in amyloidosis than we initially thought. So here in this image, a 6010 in green labels the amyloid, and then the microglia are labeled in red. And basically, there's a significant colocalization with the microglia and the amyloid, but not with the astrocytes. And this can also be appreciated in a 3D volume view with depth coding. And essentially, the colors here represent a specific depth at which the label can be detected by the microscope. And the point is that the colors match up here for the oligomers and the microglia, but they don't match up for the oligomers and the astrocytes, therefore reinforcing our initial findings just in a three-dimensional space. We also found that the basement membrane around the vasculature was reduced in the animals injected with the oligomers. And here's an image basically showing that. In red color is the blood vessel labeled with CD31. And we used the collagen antibody to label the basement membrane in white. And basically, these graphs show that there is a significant reduction of the basement membrane coverage of the cerebral vasculature here in the absence of any changes in vascular density. And this is important because the basement membrane is essentially the glue that holds the astrocytic end feed to the blood vessel cells and make sure that they can make sure they have adequate contact with those cells and can communicate appropriately. So earlier, we hypothesized that the degradation of this basement membrane could be due to the release of excessive MMPs in the body, which are normally helpful proteins for digesting foreign proteins, but at excessive levels, they can cause pathological problems. So this is essentially what normal looks like. In red is aquaporin 4, which labels the astrocytic end feed, and you can see these end feet basically line the blood vessels so well that it just looks like a blood vessel. In the experimental animals, compared to the control, we saw significantly elevated levels of MMPs, and this is what we saw by immunofluorescence staining. So in these images, MMPs are labeled in green, and the astrocytic end feet are labeled in red. And they have their respective heat maps for their individual channels, so we can see them in a different light. So essentially here, this arrow is pointing to a blood vessel that is outlined nicely by the astrocytic end feet, and it just looks like a blood vessel. And you will notice that there aren't any MMPs here, but in the where the arrowheads are pointing are the areas where there are actually higher levels of MMPs, and adjacent to those MMPs is a reduction of the perivascular coverage of the astrocytic end feet along the blood vessel. We also found that when we look more closely at the astrocytic end feet alone, we find that there's increased overall expression of the marker aquaporin 4, but actually the ratio of perivascular to parenchymal astrocytic end foot labeling is decreased, and this was interesting to us because they mirrored identically the findings found from the human brain tissue published in 2017 in JAMA. So basically what these images and graphs show is that there's fewer astrocytic end foot polarization on the blood vessels and impairing the ability of the astrocyte to communicate with the blood vessel and ensure adequate perfusion to the brain. So in summary, we provide supportive evidence that these soluble amyloid beta oligomers can enter the brain and cause reactive gliosis, whether it's interacting with the astrocytes or microglia. How much of that is happening is not necessarily clear at this point, but eventually down the line there is a release of MMPs into the parenchyma, and that causes reduced basement membrane integrity and also loss of perivascular localization of the astrocytic end feed on the blood vessel. And so future work in this regard will involve dissecting this mechanism here behind what exactly is causing these microglia to react to the oligomers and what downstream effects and consequences are there because of that. And the ultimate goal for us is to be able to prohibit the astrocytic end foot depolarization from occurring in the first place, whether that involves blocking the release of MMPs or just blocking a downstream or upstream process. That remains to be determined. So with that, I'd like to end my talk with a big shout out to my primary research mentor, Dr. Ian Kimbrough. He's been a major point of support for me and has always encouraged me to push forward throughout this entire process. Special thanks to Harry Sondheimer for letting me join the lab pretty early on in my med school career. That was very nice of him. And thank you for everybody else, Lata, Trey, and then undergrad students, Megan and Andrew Stublin, for helping me with carrying out a lot of the experiments here. Without all of them, I would not be able to be here today, so thank you so much for that. And I think my dad might be tuning in, so hey, Dad, and thank you.
Okay, we are waiting for some questions coming in via YouTube, but while we monitor the chat here, Chris, I've got a question for you. So effectively, your conclusion is that the microglia are aberrantly reacting to the soluble beta amyloid molecules, correct? Yes. So what would you speculate is the reason they're reacting to one type of soluble monomers versus normal? What is normal? So
that's a good question. I think that's something definitely that we've thought about and tried to bring into context. Like we tried to do this experiment, or a future experiment would have entailed like controlling these oligomers with any other foreign protein or a scrambled protein, because Dr. LeConte is right. Like the microglia are essentially inflammatory cells of the brain that react to or can react to any foreign protein. So it's interesting to see this sort of specific reaction to the oligomers, but I think there's still a lot to tease out there. And the literature does show specific receptors that will respond and bind specifically to oligomers. So we have a fair reason to believe that this is a specific reaction, but definitely I think there can be more work done in terms of dissecting it out and clarifying exactly what is going on between the interaction between the microglia and the oligomers.
Great, thank you. Any other questions from those in the room? No? Well, I'm not seeing any new coming in. There's a bit of a lag with YouTube, but we'll thank you again for your excellent talk. Thanks so much, Chris.
Thank you.
Hi all, my name is Tom Liu. I'm a fourth-year medical student at Virginia Tech Curlian School of Medicine. I'm really excited to present to you my project today, which I've been working on for the past 4 years or so, titled Effect of Myocardial Infarction on Sodium Channel Organization. I really appreciate all of the mentorship that my faculty mentor, Dr. Rob Gordy, has given me, and also special thanks to my collaborators, Dr. Jia He at the Virginia Maryland College of Veterinary Medicine, Xiaobo Wu and Ryan King, who are PhD students at the Fralin Biomedical Institute. The main goal of my project has really been to investigate the mechanisms behind the development of arrhythmia following myocardial infarction. Clinically, this is a serious disease that affects approximately 800,000 Americans per year. And in short, plaque buildup or some event causes a stoppage of blood flow to an area of the heart, causing an ischemic injury. In patients who survive, the scar tissue that forms causes significant alterations in the mechanical and physiological properties of the cardiac muscle, and the loss of functional heart muscle is a notable risk factor for the development of arrhythmia. The literature actually tells us that one of the major causes of post-infarct arrhythmia is due to the disordered patterning of gap junctions, which undergo a process known as lateralization. Gap junctions normally facilitate electrical signals within the myocardium via a region known as the intercalated disc. Here's an image of human infarct tissue, which was stained for connexin 43. There are actually 3 different regions. The remote region, which is pictured up here, or the region that is unaffected by infarction, is here, and you can note that the signal is arranged in a largely linear and organized pattern that is consistent with their location at the intercalated disc. This region here, known as the scar region, is largely absent of signal. And this region in the middle is a few cell layer thick region known as the injury border zone, where the connexin 43 signal is slightly more disorganized and connexin 43 is lateralized. In fact, this has significant consequences, and in this canine infarct tissue experiment, the arrows depict current direction, and the closely spaced lines here represent slower conduction. Conduction is slowed not only in the region of the scar, but also directly adjacent to the scar where the injury border zone is. At an even smaller scale, we know that the size and organization of the gap junction is regulated by a scaffolding protein known as zonuloglutens 1, or ZO1. ZO1 will bind to a specialized region on the C-terminus of a connexin hemichannel, and it will tether them to the actin cytoskeleton. Once the hemichannel is transported to the right location on the cell membrane, it can meet a hemichannel from an adjacent cell and form the gap junction. And a group of gap junctions is known as a plaque, just to give you guys a little background. And one of the things that we found in our lab is that inhibiting this interaction has actually demonstrated therapeutic potential in various situations, such as in wound healing and in scar tissue remodeling. We actually found that inhibiting the interaction between ZO1 and connexin 43 using alpha-CT1, a peptide developed in the Gordy lab, has potential therapeutic benefit. A former researcher in Dr. Gordy's lab, Dr. Michael LeQuinn, actually evaluated the effect of a alpha-CT1 methylcellulose patch and showed that disrupting the trafficking of connexin 43 delayed the lateralization of gap junctions from the intercalated disc at the injury border zone. In these images here on the left, connexin 43 is green and ZO1 is red. When we applied alpha-CT1, we see that the connexin 43 remains largely organized in a linear fashion, letting us know that it's still in the intercalated disc. Clinically, the alpha-CT peptide also reduced the incidence and the severity of arrhythmias, which was very interesting. More recently, there has been increased interest in studying the voltage-gated sodium channel, which is enriched at the intercalated disc. This super-resolution image of the intercalated disc shows us the relationship between connexin 43 in green and the 2 subunits of the sodium channel, NAV 1.5, which is a pore-forming unit, and the beta-1 subunit here in the bottom. And in summary, what we know so far is that disrupting the interaction between connexin 43 and ZO1 can reduce gap junction remodeling and arrhythmias associated with myocardial infarction. And we know that sodium channels are enriched at the intercalated disc. However, sodium channel remodeling in the context of myocardial infarction has not yet been studied. And so the objectives of our study were as follows. We wanted to determine the effect of myocardial infarction on sodium channel organization in the injury border zone, and we wanted to see the degree that an alpha-CT peptide, alpha-CT11, which is a variant of alpha-CT1, modulates the changes observed in the infarct border zone. To accomplish our objectives, we collaborated with Dr. Dia He at the Virginia Maryland College of Veterinary Medicine. Our experimental protocol involved 12 8 to 10-week-old Charles River rats who underwent an ischemia reperfusion injury. Basically, the experimenter temporarily tied the left anterior descending artery for about 40 minutes, causing a mild to moderate myocardial infarction. After restoring blood flow, half the rats received direct injection of saline or alpha-CT peptide to the region surrounding the injury. Finally, 3 rats from each arm were sacrificed at 24 hours or 28 days. The hearts were preserved and sent here to FBRI for further experiments. Once the hearts arrived here, we took a lot of effort to thinly slice the heart tissue to preserve it for both volume measurements and immunolabeling experiments. And even though I'm not going to have too much time to talk about this, I really just wanted to give a shout out to Xiaobo, who was really instrumental in collaborating with me to develop a volumetric assessment model for looking at these scars and reconstructing them using MATLAB and R. We were really able to present this at the AHA conference this past fall. So thanks, Xiaobo. A large part of my project was actually devoted to validating the novel sodium channel beta-1 antibodies in our rat tissue, and this hadn't yet been done since the beta-1 antibodies were carefully developed in our lab. After developing the protocol and validating it, we immunolabeled rat tissue and acquired single optical section images of the injury border zone, the remote region, and the scar region, using a method of identifying the area in which the quantity and quality of the connexin 43 signal was the strongest. We then developed a protocol for processing these images and consistently labeling where the intercalated discs are for future measurements. The red and green signals here represent the sodium channel, beta-1 subunit, and connexin 43 respectively. And we process these images in a way to reduce background noise. This is an example of the way that we use to isolate the different connexin 43 signals and sodium channel signals in a blinded fashion. The script which I wrote will show a series of images with varying levels of noise, and the user selects the images with the threshold level that has the least background noise. And you can see an example of the end product here. This is the original image on the left, and on the right you can see the processed image where the connexin 43 is in green and the beta-1 sodium channel subunit is in red. Before we jump into our data, I wanted to review a few measurements with you. Our measurements included puncta size, which is the measurement of individual dots of signal, which represent a grouping or cluster of either connexin 43 or beta-1 subunit. In addition, we also measured colocalization, which is a percentage overlap of 2 signals. An example of that is here, where the red and green signals overlap to make yellow. If there's less colocalization, we estimate that the objects are further apart. And if there's more colocalization, we estimate that the objects are together, potentially interacting. Finally, here's just some of our data. Although it's not shown in this slide, we observed that the overall area of beta-1 was decreased in injury border zone tissues compared to remote tissue regions. In addition, the proportion of beta-1 at the intercalated disc was decreased at the injury border zone, and you can see that in this graph down here. In fact, the decrease in beta-1 is seen as early as 24 hours. These images up top here just show the various labeling of beta-1 at different time points and different regions in the tissue. The intercalated discs are circled and have a caret, and you can see that in the injury border zone at 24-hour and 28-day time points, there appears to be more signal of beta-1 outside of the intercalated discs. The lateralization of beta-1 appears to be independent of peptide treatment, as there were no differences in the proportion of intercalated disc localized beta-1 when comparing the black and gray bars shown here, although there does seem to be a little bit of a trend at 28 days. Thus, it does not appear that the application of a peptide that inhibits the scaffolding of connexin 43 affects the post-infarction distribution of beta-1. Interestingly, peptide treatment did increase the average puncta size of beta-1 at the intercalated disc of acutely injured hearts that received alpha-CT peptide, pictured right here in the top right, suggesting that the remaining beta-1 at the intercalated disc may be closer together. In addition, the overall frequency of the lateralized beta-1 puncta was increased in chronically injured hearts that received treatment. although I'm not yet sure what to make of this data, this gives us an idea that the sodium channels that remain at the intercalated disc after an infarction may be clumping closer together when the interactions between connexin 43 and Z01 are inhibited. Finally, we took a look at the level of overlap between the connexin 43 and beta-1 subunit. The overall colocalization of connexin 43 and beta-1 was decreased at the intercalated disc, which tells us that the objects may be further apart in general. And you can see that there's an interesting trend towards the alpha-CT peptide increasing colocalization of connexin 43 and beta-1 respectively, black and gray, black and gray. Although this is only a trend, I think it's interesting to imagine that the alpha-CT treatment, which is applied only once, can have an effect on the level of interaction between connexin 43 and the sodium channel subunits 4 weeks after an injury when all the proteins have been recycled by the cell many times. And in summary, just to recap some of the work from the past couple of years, what we've really done is we've developed a number of tools for investigating peptide therapies that we might want to test in this kind of model. And some of our preliminary findings are that the beta-1 subunit lateralizes from the intercalated disc as early as 24 hours after infarction, and this does not seem to be dependent on changes in connexin 43. And we also find that inhibiting the scaffolding function of connexin 43 preserves the association of connexin 43 and beta-1 at the injury border zone. Again, these findings are preliminary. Some of the strengths and limitations of the study, our sample size is pretty small. After splitting into the groups and the time points, there's really only three rats per group. And in addition, the 2D images don't capture all the planes because each tissue can be 5 microns thick. You're only really capturing a really small segment of that. And so if two objects may be really close together, but they're in a different plane, it may be very difficult to visualize in our current model. And third, one of the big things that we realized, and this is true of all experimental myocardial infarction models, the level of experience of the surgeon can really result in the variability of the infarct quality. And so that's something we might want to address in the future. In terms of future directions, as I was saying, we do want to improve our experimental study design, improving the protocols there. And we want to potentially determine the time course of sodium channel remodeling, just because it seems like it happens as early as 24 hours, but we don't exactly know, how that happens in relation to connexin 43 just yet. And some of my personal lessons that I've really learned over the past couple of years, experimental planning is really key. I think the first time I stepped into the lab as a first year, I really just wanted to get started with the experiments. And it wasn't until very much later where I realized, even if you're not doing anything, you can spend a lot of time thinking about your experiments and making sure that they're planned correctly and save yourself a lot of heartache in the end. Learn to ask questions early. And really learn to delegate and share the work, and I think that makes science a little bit more fun. So with that, I'd really like to thank Dr. Gordy, my research mentor, and special thanks to many of the people in the lab, Jane Jordan, who really helped me with a lot of the experiments. It was my first time making a foray into the basic science world. Miao Chen, who performed some of the initial experimental MI models. Ryan King, who taught me a lot about coding and developing some of these algorithms for analysis, and the rest of the Gordy lab. And also special thanks to the American Heart Association for my student scholarship in cardiovascular disease. It really gave me a lot of confidence to continue to pursue this work throughout the 4 years. Thanks so much.
Excellent words for the students listening at home. The need to do some experiment planning, and that you can do a lot. Maybe even if you're not in the lab, you can still work on planning your experiments and doing some reading and really thinking about what you're going to do when you get in there. Great talk. We have a few questions online. So Tyler Wilson wants to know, first of all, he says, great stuff, Tom. How do you believe this research will change clinical practice moving forward?
I think that at least at this point, it might still be a little bit far. I think our next goal would be to do something like a large animal model. I think I found a lot of interest in this study just because of its applicability. And I'm hoping that, at least for me, it'll teach me how to better understand how we can take therapies from kind of the bench to the bedside. I know that's kind of really overstated and overused these days, but at least that's kind of how I think about it in my mind.
Great, and then we've got a couple other questions. So Hirsch Patolia, this is incredible and thorough work, Tom. Do you imagine this peptide augmenting post-infarction mechanical recovery of cardiac muscle?
Could you repeat that?
Absolutely. Do you imagine this peptide augmenting post-infarction mechanical recovery of cardiac muscle?
I think that that's certainly possible. One of the prior studies did show that there's less incidence of arrhythmia and a decreased incidence of the severity of the arrhythmia. And I think that the peptide has also been shown to reduce scarring. And so in certain situations. And so I think that if it does reduce a lot of the scarring, and scarring is a big determinant of myocardial function, then they should go in hand and it would help.
Great. I'm going to take the liberty of asking one of my own questions. So you highlighted the fact that there was an impact of the peptide administration 28 days later. Can you speculate, and I know I'm asking you to stretch here because I'm not sure it's really known, but what might be the mechanism behind that long-term impact with a single dose of this peptide 28 days prior?
That might be a little bit above my level of personal understanding, to be completely honest. But I would say that it seems that there are a lot of treatments out there that, really you give once and can change like the structural composition of a cell, or kind of really set up the cell to change differently. I'm thinking about one of the experiments that a former PhD student in the Gordy lab did, looking at scar and wound healing, and application of the peptide just early on in the treatment really reduced the level of fibrosis and scarring in the skin tissue. And so it could just be that it's maybe even like tethering some proteins somewhere, or keeping proteins in a certain location that long-term will really benefit and reduce the development of scar tissue.
Excellent answer. It's a tough question, I know. And then last question from Kermit Zhang. Awesome talk, Tom. I saw a lot of the colocalization data in your experiments. Did you or your mentor ever actually look at interactivity between connexin 43 and the sodium channels at all?
In my project, we did not. There's been a lot of other studies going on that have really looked at the interactions between connexin 43 and the sodium channel. Actually, a number of years ago, they used a staining process called dual-link staining that basically really tells you when two proteins are within like 50 nanometers apart. The resolution of the confocal imaging, I think the limit is maybe like 200 nanometers, and so it doesn't really tell you that, but based off of the previous data and some of the other work that Dr. Varagavan, who's now at Ohio State, is doing, show us that these two proteins are very intimately associated.
Excellent. Thanks so much. Appreciate it, Tom.
Thank you all.
Okay, everybody, welcome back to the second session of the Class of 2020 VTCSOM Research Symposium. We are here to hear several more presentations from our Letter of Distinction in Research Scholarship winners. And our first speaker of this session will be Mercedes Robinson, who worked with Dr. Pearl Chu of FBRI.
All right, thank you, Dr. LeConte. My name's Mercedes. I'm a fourth year medical student at VTC, and my mentors are Pearl Chu and Vanessa Brown. And I will be presenting for you all my project, which is titled Gender Differences in Reward and Punishment Processing Within Depression. So just some background on depression. Major depressive disorder is characterized by low mood and or loss of pleasure in daily activities for more than 2 weeks. And this change in mood represents a change from the person's baseline. Major depression affects an estimated 300 million people worldwide, and it's a leading cause of morbidity and mortality. And as such, there is a gender disparity noted in depression and many other psychological disorders, where women are nearly twice as likely to experience major depression, have more frequent episodes, and a greater number of depressive symptoms. One of the core features of depression is cognitive dysfunction, and something that my lab is interested in studying is decision making, in particular the difficulty with decision making that happens with depressed patients. Individuals with greater depressive symptoms make less productive decisions, and they engage in fewer adaptive decision-making strategies. And major depression is caused by a variety of environmental and biological factors. And one of the models proposing how anhedonia arises, which is a core feature of depression, is through dysfunctional interactions between stress and brain reward systems, which we're particularly interested in studying. So to that effect, we use reinforcement learning models to study these dysfunctions that arise in reward learning. And these reinforcement learning tasks quantify how people use positive and negative feedback to make decisions. The ultimate goal with reinforcement learning tasks is to learn through trial and error how to maximize your rewards or positive outcomes that occur, and to avoid punishments or negative outcomes, because actions that are followed by a reward are more likely to be repeated in the future. Depressed patients show dysfunctions with reward learning, such that they have hyposensitivity to reward and maladaptive responses to punishment, and also have difficulty with modulating and utilizing feedback to be able to change their behavior appropriately. And something that I'm interested in with my project is to see if these dysfunctions or these alterations are different between depressed men and women. And so just a little bit of our methods and participants. So we had a total of 151 participants. The ages range from 18 to 55, about 65% were female. We had 53 non-depressed controls, as well as 98 in our depression group, and they met DSM criteria for major depression or dysthymia. And they also had a score of greater than 12 on the Beck Depression Inventory Scale, which is equivalent to a mild depression. So a little bit more of our methods. So participants went through a screening visit on their first visit, where they filled out the consent, completed the structural clinical interview where they received the diagnosis of depression, and then filled out a variety of questionnaires and assessments. They then came back for a week later, where they repeated those questionnaires, but also completed an fMRI scan, where at the same time they were doing a probabilistic learning task. And this is the reinforcement learning task that we use in our study. And basically with this task, the participants view abstract stimuli, and they make specific choices associated with monetary rewards and losses. And this probabilistic learning task is really important in measuring how sensitive an individual is to positive and negative feedback. Just to explain a little bit more about the task, so participants are presented with two choices with either a gain or loss block. Gain is when they're responding to rewards, loss is when they're responding to punishments. One of the choices has a higher probability of leading to the better outcome. So in the gain block, that means a larger reward or a 75-cent gain. The other choice has a higher probability of leading to the worse outcome, which is a smaller reward in the gain block of 25 cents. And just to show you what that looks like in the loss block, the choice that has a higher probability of leading to the better outcome would then be a smaller loss, because participants want to maximize the amount of money that they can gain in this task. And so in the loss block, that means just a 25-cent loss for making the correct choice. And the other choice, again, with the worse outcome, would be a larger loss in the punishment block. So participants complete a minimum of 50 trials, and they're encouraged to choose the pattern most often associated with a better outcome. So it's a probabilistic task. It means that even though participants make the correct choice, they will not always be rewarded. And this parallels what happens in the real world, where we make choices every day, but we know some choices are right, some are wrong, and even though we make the right choice, we're not always rewarded for the choices and decisions that we make, but we stick with them and we keep making those choices because we know that for the most part they're most often associated with a positive outcome. And so the participants also, we use an adaptive algorithm, and basically this balances performance differences across participants, because what we're really interested is in the learning and not the overall measures of their task performance. And just to segue into that, we first looked at these overall measures of task performance. So looking at the choices people are making, and if they decide to stick with the same choice or switch their options. And on the graph to the left here, let me just change that so I can, my laser, there we go. We have proportion correct. And you can see that the cross group, whether they're depressed status and gender, that we don't have any significant differences in the proportion that participants answer correctly. And same with switch, in the proportion that they're switching, we don't see any significant differences across group or gender. And so this is why we wanted to continue looking at trial by trial data to see, again, to explore how people are learning through trial and error and the choices that they make. Are they able to utilize the positive and negative feedback that they're getting in the task to then change their behavior and change the decisions that they make? All right. And so we looked at trial by trial data across both the gain and loss conditions, and we did this by using hierarchical logistic regressions. And the measures of task performance, again, as I mentioned, are choice, so whether the participant made the wrong choice or they made the better choice. And whether the participant switched their options or they stayed with the same choice. And we look at these things because it allows us to see, how sensitive an individual is to positive and negative feedback. For instance, if a person makes the correct choice, but they end up getting the worst outcome or a larger loss, they could still stick with that in the next trial because they know that it's better in the long run, that they'll be rewarded for making that decision. Whereas someone that's more sensitive, if they get that larger loss, they are more likely to switch their choice and be hyperactive to that. And so with our model, we estimated the likelihood participants would make the better choice and the likelihood that they would switch options. And the predictors in our model were gender and previous trial outcome. And so on our first graph here, we're looking at just depressed participants first. And on the y-axis here, this is our likelihood estimate. So this is the coefficient of the predictors in our model. In particular for these, this is for the previous trial outcome or their most recent feedback that the participant received, predicting the choice and also the likelihood that they would switch on the next trial. And so we can see here that when responding to rewards in the gain condition, that there is a significant interaction between gender and previous trial outcome for choice, such that depressed men are much more likely to make the appropriate choice if they receive the better reward on the previous trial. We don't see a difference for choice when responding to punishments in the loss condition, and we don't see any significant differences between depressed men and women, whether with regard to the likelihood that they would switch in the gain condition. But we do see a significant interaction when responding to punishments such that depressed women are much more sensitive to punishments in that they are more likely to switch if they receive the worst outcome on the previous trial. And just to show this interaction a little bit more between the previous trial outcome and gender, we have just here a graph or interaction plot showing the choice probabilities in the gain domain. And you can see here that overall, when people receive the better outcome, they're more likely to make, the probability that they'll make their appropriate choice is higher than when they receive the worst outcome, but that the probability is even higher for depressed men that they will make the correct choice when they receive the better outcome on the previous trial. And then to show that interaction between switch behavior and the loss condition, we have another interaction plot here that shows that, again, when overall participants, when they receive the worst outcome, or the probability that they will switch is higher than when they receive the better outcome, but that difference is enhanced even more with women. When they receive the worst outcome, they're more likely to switch their options, which again suggests that they are sensitive to losses compared to depressed men. As comparison, we did not see any significant gender differences with our non-depressed patients across all domains, with choice behavior and also with switch. And some of the conclusions for my study, so our results suggest that depressed men choose better on the task when feedback is positive, while depressed women are more sensitive to negative feedback. We did not see any differences in overall task performance between our non-depressed control men and women. And some additional analyses that I did not have time to talk about were that depressed men are more likely to respond faster when they're rewarded or they receive the better outcome on the previous trial, and that task behavior is also influenced by depression severity and anhedonia. So some implications for my project: understanding decision-making processes of depressed individuals and the role of gender can help to provide insight into the etiology of depression. Depression is a very heterogeneous disorder, and it can present in a multitude of ways and influence people. And I think the more that we can better understand the etiology and the cause of it, the better that we can improve people's lives. And for instance, with depression, bad decisions and negative experiences from them may reinforce an already negative view that depressed patients hold and serve to generate feelings of guilt and hopelessness. But not all is lost. Depressed patients are able to learn or gain back the ability to make appropriate decisions. And we've shown that in our work that these alterations in reinforcement learning, reward learning, correlate with symptomatic improvement, and that teaching decision-making techniques as an adjunct to behavioral therapy can serve to help people because depression, although it's a treatable disease, it does have a remission rate of 50%, and so I think there's a critical need there to explore other options for being able to successfully treat people with depression. Just want to acknowledge my mentors again, Dr. Pearl Chu and Dr. Vanessa Brown, and all the members of the CKC lab, and Katie McCurry as well for their support and guidance on this project. Take questions.
Great. We've got a question in the room. We'll go ahead and take that one first.
We do indeed. Thank you, Mercedes. It's a pleasure to hear your talk about depression and the gender differences. Two questions. The first has to do with the magnitude of differences. You mentioned they were statistically significant, and we saw some graphs that showed a lot of overlapping distribution. How large were the gender differences? And then my second question is whether, as you interpret the interaction effects that you found and the differences, do you think they attribute more to the biological, this is like the hardest one, the biological or the socially determined aspects of gender?
Okay. To answer your first question about the magnitude of the effect, so in our model with logistic regressions, you're basically estimating a likelihood of a behavior. And in this instance, it's whether people will make the more appropriate choice and if they will switch. The reason why I showed that one graphic there with the probabilities, there is some overlap, but that's because it's a different model or different way of estimating probability, similar to how we estimate likelihood with our logistic regressions. And so the reason why I like to keep that graph in there is just because it shows just the overall trends. And so even though there is overlap in the variance of the probabilities for those, the magnitude is that for each of those interactions that we found in our specific model, they were statistically significant. And so I hope that serves to answer your question. I think it's just the way that the variance is captured and how probability is estimated, why you see, in our model there is a significant interaction, but then with the specific plot that there does appear to be overlap there.
It can be hard with beta coefficients to quantify the magnitude of the association.
Right.
So I appreciate that statistical significance is often used as an indicator. And then about more of the conceptual model question of what does it mean back to the biological versus social determinants.
Yeah. I think it's, I think that's why depression is so fascinating, given how prevalent it is and the fact that it is heterogeneous. and that the etiology, although we know that there are many risk factors associated with depression, we can never fully just pinpoint it on one causal factor that then will subsequently lead to the development of depression. I do think in terms of the role of this project in understanding that etiology, I think there is an instance of just the impact of behavior. And so what we're studying right now in this project is how people, the decisions that people make and how it can contribute to a vicious cycle that people undergo in depression, where they're continuously making bad decisions or they're avoiding making decisions as a coping strategy, which doesn't further their interests and contributes to a lot of the hopelessness, the guilt, missed opportunities in their lives that just serves to exacerbate their experience. And I think separate from just the social and biological, I think that there is an environmental and behavioral component, and that we have shown that in our work that if you modify and manipulate people's decisions and to help them make more productive decisions and that further their interest, that you do see symptomatic improvement with that. And so I think that for me is the clinical significance of this project specifically in terms of focusing on that in therapy and potentially even driving targeted therapy with men and women, for instance, focusing more so on positive outcomes that happen in men's lives and maybe trying to redirect women from focusing so much on the negative outcomes that are occurring in their lives.
We have a lot of great questions from YouTube. I'm going to ask one from Nina Harding. Very interesting study, Mercedes. Do you think your results would be affected if the rewards/punishments offered in the study were different? For example, larger payout, cupcakes instead of money, etc.
Yeah. Well, cupcakes would certainly be effective, at least for me, a motivating factor. I think that the reason why we use this specific task is that it specifically activates those reward processing pathways in the brain and dopaminergic transmission. And so, our task is basically an adaptation of previous tasks that have shown to activate those areas and those reward processing and brain reward systems. And so I think that the money, it's such a small amount that it's not necessarily a huge factor in the sense that it's going to drive behavior so much, so that like if we increase the amount of the payout, that would change their behavior. I think that the amount that we provide is enough of a motivator in the sense of it's a positive or a negative outcome, and that then in itself can influence people's decisions.
Intriguing study. Thanks so much for answering
you.
questions and presenting to us today.
Thank
Thanks.
you.
Dr. John Chappell, sorry, John, from FBRI. Thanks so much for joining us, Harsh.
Good morning, or good afternoon, everybody. Thanks for tuning in on this beautiful sunny day. I'll be presenting a lot of the work that I performed with the Chappell lab, as well as the Shane Pierce Kotler lab, involving the creation of an agent-based model that simulates pericyte migration and recruitment to the angiogenic front in response to PDGFB signaling. So endothelial cells and pericytes are remarkably important players in angiogenesis and vasculogenesis. With respect to angiogenesis, the whole process of angiogenesis, or the formation of new vascular networks from pre-existing vascular networks, starts with VEGF, or vascular endothelial growth factor A. And an extracellular gradient of vascular endothelial growth factor A, or VEGF, will actually entice endothelial cells to take on a more tip cell phenotype, therefore becoming more vascularized and forcing the creation of new lumens. This occurs when VEGF actually binds to VEGF receptor 2, a tyrosine kinase receptor that is present on the membranes of all the endothelial cells. Once there has been sufficient activity and activation of the VEGF receptor 2, the endothelial cell will actually fully transition to a tip cell phenotype and therefore upregulate its extracellular secretion of platelet-derived growth factor B, or PDGFB. This chemokine is primarily functions to actually recruit pericytes to the angiogenic front right here, such that pericytes will start to be recruited to this site and serve kind of two functions. Number one, they serve to stabilize the very delicate angiogenic front, and number two, they serve to ensure that there is a lack of microhemorrhage at the vascular front as well. This very delicate and concerted process and signaling among different chemokines and receptors is something that can be very easily simulated. And by the way, just in this figure, endothelial cell is represented by EC and PC represents pericyte. And as I mentioned, as this is a very simulable process, previous studies have actually demonstrated that an ABM can appropriately, or agent-based model, can appropriately simulate the interplay between endothelial cells and pericytes. And this was what our collaborators had developed at University of Virginia with Dr. Shane Pierce-Kotler and Dr. Walpole. And they demonstrated that a paucity of pericytes at the angiogenic front will actually negatively affect vascular patterning and adversely affect the stability of the angiogenic front. However, this model lacked any consideration of pericyte migration. In that model, pericytes only serve to inhibit laterally the formation of new bridges from pre-existing bridges of endothelial cells. And right here is a very beautiful ex vivo image / video that demonstrates kind of the vascular formation at the angiogenic front. I want to bring your attention to the bottom left of this screen, where you can actually see an endothelial cell stalk in green creating a bridge, and red indicates pericytes, and they trail behind this vascular tip. Therefore, likely secondary to the recruitment of pericytes at the angiogenic front in response to PDGFB. And I mentioned agent-based modeling earlier, or ABM, and how we had a pre-existing model using ABM to simulate the behavior of pericytes and endothelial cells. So what is agent-based modeling? Well, agent-based modeling is a very interesting modality that allows users with the ability to examine how individual behaviors and microscopic processes can actually affect the emergent phenomenon and the macroscopic behavior of a system. So what users can do is effectively impart rules or immobile agents upon the mobile agents or the individual processes in the world or the simulation world, and program these rules and capture these rules to steady state, or capture the phenomenon to steady state. One very interesting and the one kind of platform that we used in this lab was something known as NetLogo. It's an open source modality primarily that functions for ABM, or agent-based modeling, and it's a very easy to use open source modality, and here's a sample image from their open source website where they utilize ABM to capture the emergent phenomenon of a sheep-wolf system. So therefore, the specific aims of this project were to take that agent-based model that I had mentioned earlier and revise it in a way that we consider pericyte behavior, namely migration, within the context of PDGF signaling. So number one was to determine, is it even appropriate to include this signaling access in this model? How does the addition of chemotaxis secondary to extracellular PDGF, how does that actually affect the migration of pericytes in comparison to randomized movement? The second aim was to implement binding rules and chemokine rules that moreover reflect biology in terms of how PDGFB and its appropriate receptor bind in the model. So that involved the implementation of literature-inspired rule sets. And lastly, but probably most importantly, is to take what I'm describing as an in silico model, but then take it back to the biology and validate it with in vitro studies. And that's exactly what I'll present to you today. So the hypothesis here is that pericyte coverage, which is a laboratory proxy for the amount of pericyte stability and envelopment of the vascular network, that value will decrease in a simulated VEGF gain of function environment at a difference in our simulation that is statistically equivalent to what we had demonstrated with in vitro studies. So kind of the workflow of my model is that number one, the model involves the creation of a VEGF gradient. Recall that endothelial cells can't really do much without this gradient in place. The second step is to seed the initial cells. In this step, the user can pre-select where they want to seed the ECs, endothelial cells, and the PCs, the pericytes, in the world. And I'll kind of show the formation we use for our validation in subsequent slides. And then the most important is probably cellular movement. And cellular movement is governed by the interactions between endothelial cells, as well as the chemotaxis of pericytes to this extracellular PDGF gradient that's been created by endothelial cells. But just to kind of expose more of the granularity of this process, I'll show you a cartoon of how this actually occurs in the model. So endothelial cells will constitutively produce PDGF in the model. So they're always secreting it in the model. However, once the endothelial cell has had enough activation from VEGF with that VEGF receptor 2 I mentioned in the first slide, it will take on a yellow or a tip cell phenotype in this model. With that transition to the tip cell phenotype, PDGF will selectively be upregulated into the extracellular matrix. The assumption now is that the pericyte will then migrate to the vascular front, or the tip of the tip cell. And that right there is perhaps the part and parcel of how this model is working. And some of the more important granular global parameters of this model include the simulated area that we simulated in the model. So the whole simulation space spanned about 1.31 square microns, and every patch or pixel was comprised of 5 micron pixels in 2D space. So note that this model was in 2D. Every endothelial cell and pericyte was forced to maintain a connection, and every area of each cell was 500 microns always, and that was a constant. And every time step we simulated was 10 minutes long. And during those time steps, that's when we implement the binding rules, when we actually implement what's known as the modified Gillespie binding algorithm to actually examine how this binding reaction will occur to steady state within a 10-minute interval. And then lastly, all endothelial cells and pericytes were forced to maintain connections in the model. And right here, I won't get into the specifics, but right here are some of the specific rules that we incorporate in this model to assure users that the rules that we were using in this model are inspired by literature. These are studies that have validated a lot of these parameters. And first, to address the first aim, whether or not randomized motion of pericytes in comparison to chemotactic behavior of pericytes in response to PDGF was even appropriate to examine. Well, right here we demonstrate in the first bar graph up there that pericyte coverage, when we even just preliminarily programmed in a rule that governed pericyte chemotaxis in response to a PDGF signal, actually improved pericyte coverage. And as I mentioned earlier, pericyte coverage is our laboratory proxy and simulated proxy for pericyte stability of the vascular network. And right here is a sample image that shows over 350 minutes how that morphology of the endothelial cells and pericytes changed. I also want to bring your attention to this time graph right here that demonstrates that around 300 minutes, when a lot of these endothelial cells are taking on their tip cell phenotype or becoming yellow in the model, there's actually a global upregulation in PDGF activity. So that addresses the first specific aim. I also wanted to show this heat map that demonstrates the simulated setup we use for the validation phase of this study, in which we recapitulate a capillary formation at the angiogenic front. And these heat maps demonstrate the quenching of VEGF signal in response to binding of these endothelial cells by VEGF receptor 2. And I also want to bring your attention to these heat maps involving PDGFB as well as the receptors, and indicate that the binding occurs primarily at the pericyte endothelial cell interface. And that is even biologically, we've demonstrated that the binding of these chemokines and the interactions of endothelial cells and pericytes occur at that interface. This right here is the control kind of simulation in which we demonstrate that in the absence of a VEGF gain of function scenario, there's appropriate vascular patterning and appropriate pericyte migration to the vascular front. In terms of the transition to tip cells, only by the end of the simulation, only 3 tip cells, or only 3 endothelial cells have transitioned to a tip cell phenotype. And this simulation was performed 200 times to ensure appropriate repeated trials. And then in the gain of function model, we demonstrate complete vascular disarray and patterning, which was observed in our in vitro studies. Note that a lot of the endothelial cells early on in terms of the experiments have transitioned to tip cell phenotypes, likely secondary to an excess of VEGF into the simulated world. So I've shown you that there's complete disarray and chaos within the vascular patterning and a VEGF gain of function and a control environment, but qualitative observations are just not sufficient to validate the claims I've made early on. So that's why we take it to the next level. We use numbers. And right here, the top graphs indicate the results of the in vitro studies that Dr. Darden had performed to assist in the validation of this study, and the bottom row indicates the outputs of my simulation. So we'll start with the top graph. The top graph indicates the pericyte coverage and the lack of pericyte coverage that Dr. Darden had observed in her studies involving the titration of VEGF, or the gain of function scenario, in her studies. And she showed that there was a diminished pericyte coverage in that experimental setup. The next thing she had done was actually normalize the pericyte coverage in the gain of function model to the wild type, or the control setup, such that the gain of function coverage was a fraction of the wild type coverage. Using Dr. Darden's 95% confidence intervals, the extrema of these 95% confidence intervals, I was able, with the assistance of Dr. Alison Teggy, to create a 95% confidence interval for the simulated data, such that we have a minimum and a maximum bounds of what would be a 95% confidence interval for the validative part of my study. And as you can see, and this is for the normalized, such that the normalized coverage, such that the gain of function pericyte coverage is a fraction of the wild type. And it's quite evident here that the gain of function coverage falls within the 95% confidence interval bounds that we had determined from the in vitro studies. So the conclusions show that, the study showed that the program chemotactic behavior regarding our first aim demonstrated increased pericyte coverage relative to randomly generated movement. In our validative phase of our study, we demonstrated that simulated pericyte coverage was lowered in a simulated gain of function environment regarding VEGF. And this was statistically significant. The difference was statistically equivalent to what had been observed in vitro studies. Therefore, we preliminarily demonstrated that pericyte and endothelial cell interactions can be reasonably modeled using agent-based modeling. Furthermore, we also developed an image analysis tool to examine 3D data sets with the hopes of further taking our validation to the next level with confocal data, though unfortunately we were not able to use InSight, as we call it, in this study, but we hope to use it to examine other experimental data from our lab. Some of the challenges and future directions of this project, namely the challenges, include that I assumed when I crafted this model that the relationship between VEGF-R2 activation and the secretion of PDGFB by endothelial cells was a direct relationship. That was an assumption, and no studies have been performed to determine what that relationship is. It would be very helpful and beneficial to do a study or perform a study in which we can actually quantify that relationship so that this model can take that and apply it. The second is that this model, as with many models, simplifies the intricacies and the granularities associated with pericyte migration and endothelial cell signaling. And furthermore, we have also demonstrated that soluble PDGFR beta is actually shed by injured pericytes in response to hypoxic conditions. And we presume that this might be as a signal to diminish pericyte recruitment and increase vascular instability at that site. More simulations were possible, but unfortunately hindered by the ergonomics of the output from NetLogo. So we hope to perhaps create a tool where we can import large amounts of data from NetLogo and reasonably analyze them. At the end of the day, what we've created is a tool that is not only to validate the biology that we observe in experimentation, but also to inform new hypotheses, create new experiments, and in a reasonable manner, such that we are stewarding our hypotheses appropriately. I want to thank the following individuals: Dr. John Chappell, of course, for not only his guidance in my research, but also incredible professional guidance over the past 4 years. Dr. Pierce Kotler and Dr. Walpole for creating the initial build for this model, and Dr. Ricard for her assistance in terms of the hypothesis generation involved with this project, and Dr. Darden, of course, for providing the in vitro studies that we needed to validate the results of the simulation, and Dr. Teggy, of course, for her assistance with the statistical analysis. I also want to end with a quote from Winston Churchill, in that these times are strenuous, but success is not final, failure is not final. It is the courage to continue that counts. Take questions.
On now? Yes, okay. Great talk. You and I think, share a love for computational modeling, mixing modeling with experimental results. So this is a project that I've been watching with interest since we first talked about it. I'm waiting for a few questions to come in, but while I have the mic, I will take the liberty to ask, what are some things that you think you're going to be able to do, or what could you do in the future with your simulations that would guide experiments? How is this going to make the experimental process easier?
Absolutely, yeah. Thank you so much for that question, Dr. LeConte. I wish I had addressed that earlier. And absolutely, the way we, Dr. Chappell, envision that this platform can be used by experimenters and scientists is that we want to take a knob, let's say for VEGF or for PDGF, and just turn it all the way to the right or all the way to the left and examine how does this system behave in a long-term scenario. And then, using that data, we can create, the morphology changes this way. We can create a hypothesis from that, and then appropriately create our experiments to see, all right, are we observing that this is what we had observed in the model? So at the end of the day, I think it's to observe the system at an extra month, if you will, and then take what we find in the in silico model and apply it to the in vitro, in vivo, ex vivo models.
Excellent. Excited to see where this goes. Let's see. I'm waiting to.
While you're waiting.
Yeah,
sorry.
Great.
I was going to ask a question along similar lines, and it had to do with where this goes in terms of human health and disease. So, is this model robust enough now, or do you see it being robust enough to even look at drug targets to block tumor angiogenesis? Is that where we're going with this? Because it's cool in its own way in terms of facilitating experimentation, perhaps being more efficient in the use of laboratory resources in using this model instead. But where do you think this can go?
Absolutely. Thanks for that question, Dean Learman. So absolutely, the introduction of cellular chemokine inhibitors was something that Dr. Chappell and I had discussed early on, actually, is that this could be a platform for scientists to examine the introduction of a VEGF receptor inhibitor, perhaps, or even a PDGF inhibitor. And in terms of human health and disease, I had not mentioned this earlier, but pericyte dysfunction has been implicated in a number of pathological entities. My colleague Anisha Chadha mentioned the possibility of it being implicated in preeclampsia, but we have definitively implicated it in pathological entities such as diabetic retinopathy and neonatal hypoxia, causing germinal matrix hemorrhage and therefore intraventricular hemorrhage. And we think, and we were hoping, to utilize and apply this model to perhaps recapitulate those pathological states and introduce pharmacologic interventions in an in silico manner that can, in which we can examine how the emergent phenomena result from those inhibitions of those pathways or the activation of those pathways. Yeah.
We've got a lot of good questions on here, and a lot of people saying they like your quote, so excellent quote. One from Meeta Prakash: how does agent-based modeling distinguish functionality between the extracellular and intracellular signaling?
Thank you so much for that question, Meeta. So it's all based on the patch-wise processes involved in the model. So I don't want to get into the nitty-gritty of how the ABM itself, but ABM requires, in this specific model, requires the constituent patches of every cell to interact with each other. So the way that happens is that these patches are given kind of a sign. If a patch is has a cell sign, it's indicated as an endothelial cell or a pericyte. And using that data, the signs of these individual patches, we can actually force the patches on a cellular level or an extracellular level, because assure you, I actually have signs in the model that indicate that there's an extracellular space, that indicate that this is extracellular space, this is intracellular space, this is membrane-bound space. and we can force the interactions to occur only in those 3 areas, or one of those 3 areas, or 2 of those 3 areas. And that's kind of how I address the notion of intracellular signaling versus extracellular signaling.
Great. Thanks so much.
Thank you.
Okay, thanks for waiting for our brief pause. We're going to go ahead and hear from Mustafa Rashid, who worked with Dr. Ken Astrotech, who's formerly of FBRI.
Great. Thank you. Good afternoon, everyone. It's a privilege to be here, and thank you for being here either in person or virtually. My name is Mustafa Rashid. I'm a member of Dr. Astrotech's lab. He was formerly a member of FBRI and is now currently over at The Ohio State University. I'd like to thank him and Dr. Bharat Strickmore, along with the rest of my lab mates, for helping me throughout this project. The title of my project is The Role of the Icaro Zinc Finger Transcription Factor EOS in Th2 cells. Before we dive into EOS and Th2 cells specifically, I would like to take a step back and talk about T cells in general. The T cell population has many subsets, ranging from TFH, Th2, and the rest, as you see on the board, that all have very specific jobs or niches within our immune system. And in order for these cells to exist, they go through a pretty common pathway. It starts with a naive T helper cell coming in contact with an antigen-presenting cell, and then based off of the cytokine environment, that will induce a specific STAT transcription factor that will in turn induce a specific lineage-defining transcription factor, such as GATA3 for Th2 cells, and that will in turn cause that phenotype of that specific T helper subset to exist. So, for example, Th2 cells will need an environment of IL-4 that will induce the activation of STAT6. That will in turn cause the induction of the GATA3 lineage-defining transcription factor. This allows Th2 cells to perform functions such as fighting off extracellular parasites and helminths, such as the worms that you see above. In addition to this, Th2 cells have also been implicated in many hypersensitivity disorders, ranging from 1 through 4, including your common everyday pollen allergies to your allergic reaction due to a bee sting. They've also been implicated in reactive airway disease and asthma, along with multiple other autoimmune disorders, such as Hashimoto's, SLE, celiacs, and multiple sclerosis. So, understanding this clinical correlation of Th2s and how they come to be is important, so that way we can create targeted therapeutics in order to fight off these autoimmune disorders. For example, in MS, there's a drug known as glatmir, or myelin basic protein, that actually will shift a Th1 heavy population in patient to a Th2, and that is able to allow for symptomatic control in these patients. So being able to further understand this network of how these cells come to be, we can have more targeted medications for these patients. So that being said, I'd like to dive a little bit deeper into how these Th2 cells do come about in some of the common pathways. Again, we start with a naive T helper cell and coming in contact with an antigen-presenting cell along with specific cytokine environment. For Th2s, one of the most common or notable pathways is through IL-4. The IL-4 cytokine will come in contact with the IL-4 receptor. That receptor will cause the dimerization and phosphorylation of STAT6. That STAT6 will then translocate into the nucleus and cause the induction of GATA3, and that GATA3 will in turn lead to the phenotype of the Th2 cell and the cytokines that it can produce. In addition to this pathway, the IL-2 pathway is also a common pathway. IL-2 will bind to its cognate receptor. IL-2 actually leads to the phosphorylation and activation of STAT5. STAT5 also will translocate to the nucleus, and that will in turn cause increased expression of the IL-4 receptor, creating a positive feedback loop. All of these in turn leads to the Th2 profile, which includes production of cytokines such as IL-4, 5, 6, 10, and 13. IL-4 is key for IgE production. IL-5 is key for IgA production by B cells. IL-6 is great for recruiting eosinophils, and IL-10 is needed for inhibiting interferon gamma, and IL-13 does a little bit of mixture of those previous cytokines. So in addition to these key players, the last decade or so, the IKZF family members has also come to be known as key players in lymphopoietic tissues and differentiation of immune cells. They include 5 family members ranging from IKZF1 to 5. IKZF4 is EOS, 1 is Icaros, 2 is Helios, 3 is Aeolus, 5 is Pegasus. For the purpose of this project, we'll be focusing on IKZF4 and EOS. These members of this family have all been shown to have conserved sequences and binding motifs, so they all do have a protein-protein binding motif in the C terminus, along with DNA binding motifs in the N terminus. our lab has shown that some of these IKZF members and STAT factors do have a relationship and are important for the differentiation of specific T helper cells. Specifically, we've shown that STAT3 along with ALOS are able to form a complex in TFH cells and are crucial for their formation. In addition to that, current research in our lab has shown that STAT5 and EOS interact in TH1 cells are important for TH1 cell differentiation. So this begs the question, do IKZF factors have a function in the expression of the Th2 gene program? Specifically, we hypothesize that the IKZF family member EOS regulates the expression of the Th2 gene program through a cooperative regulatory mechanism with STAT5. So to begin, Th2 cells were cultured and naive T helper cells were cultured in Th2 polarizing conditions, and we wanted to determine which of these family members was the most highly expressed in Th2 cells. In order to do that, we isolated some RNA and used cDNA and QRT-PCR to determine the expression levels of the 5 family members in Th2 cells. Interestingly, we saw something that is pretty well known in that IKZF1 or Icaros is highly expressed, which is highly expressed amongst all T helper cells, or the majority of them. In addition to that, we saw that EOS was highly expressed as well. And this was all done through a ANOVA analysis showing that Icaros and EOS were the 2 that were most significantly expressed. Taking that a step further to confirm that EOS is highly expressed and that it is different than other T helper subsets, we compare that to other subsets such as Th0 and Th17, and we do indeed see that it is significantly expressed compared to other subsets. So now that we see that at the genetic level and mRNA level it is highly expressed, the next question was, well, does this translate to the protein level? In order to answer this, we utilized flow cytometry using anti-EOS antibodies that allow us to determine the amount of mean fluorescence, or that translates to the amount of protein that are in those cells. And this is the example of those results. We can see here with the orange line in our histogram that EOS has the highest fluorescence intensity in our multiple subsets of Th0 and Th17. And this is the sum and average of all of those. So we have shown that it is there at the genetic and the protein level. Now we want to understand how or what its role is. In order to do that, we looked at the target genes of EOS. So to answer this, we cultured our T helper 2 cells with a silencing RNA that silences out Icaros, and then we performed QRT-PCR. So here we see the white is our control, which is our normal, and then our SI-IKZF4 is in black, which is where our IKZF is silenced. So here we see about a half reduction, or a half-fold change in our IKZF. And then we see that it's specific for IKZF4 and that the rest of the IKZF family members have not changed. Moving forward, we see that GATA3, the lineage-defining transcription factor for EOS, is not affected. So this leads us to hypothesize that EOS is functioning independent of that GATA3 lineage-defining transcription factor. Moving forward, we looked at some of the other key genes of EOS, and IL-4 and IL-13, as we mentioned earlier, are 2 of the key genes for Th2 phenotype. We do see that IL-4 and 13 are both reduced in expression. We also looked at PRDM1, which is a transcriptional repressor, in that it silences other genes, which is key for Th2 differentiation, had no change, and there was no change in the IL-2 receptor alpha expression. We were able to then obtain some EOS knockout mice and perform some of the similar assays of QRT-PCR, and here we can see the blue is our wild type, while the red is our IKZF4 or EOS knockout mice. Here, in this first graph, we're able to see that our mice are indeed IKZF knockout mice, and that we see that there is no red or no IKZF4 expression in these mice. Then we looked at similar genes again. Again, we saw no change in GATA3 expression. We did see decrease in IL-4 expression. Again, we see that it's significant at the 72-hour time mark, and we can appreciate that it is decreased at other time marks as well. We also see that there is, again, decrease in IL-13 expression. And moving forward, we looked at IL-2 again along with receptors. We see an increase in IL-2 expression, but we see a decrease in the receptors for IL-2. And then we also are able to appreciate PRDM1 being decreased in expression levels in our cells as well at the 72-hour time mark. Some of the changes between our EOS knockout mice along with our siRNA data could be due to the fact that our sRNA data showed a half-fold decrease, while our EOS knockout mice had no EOS present at all. So taking this a step further, showing that there are target genes that are affected by EOS, we wanted to determine how this could be, how could this come to be about. Previous research has shown that that EOS can bind to DNA with those DNA binding motifs, or possibly with STAT factors. In order to do this, we utilize chromatin immunoprecipitation, which allows us to essentially look at segments of DNA and using antibodies to see if our protein is there. And we looked at two regions, a PRDM1 enhancer region, along with a negative control. And looking at that region, we know where STAT5 normally binds, and we look to see if EOS is bound there as well. And our data indeed does show that there's both STAT5 along with EOS bound at that positive enhancer region. And we wanted to determine if this had any downstream consequences. So we looked at the protein product of PRDM1, which is BLIMP. And so indeed, when there's no EOS present, we are not able to have any of this DNA be translated, and so therefore no protein is created. So here we have our EOS positive mice where we see BLIMP, BLIMP1, and our EOS knockout mice where we do not see it. Moving forward, so we were able to determine that this is possibly due to DNA binding or possibly due to protein binding. So in order to determine that, we utilized mutant cells and transfected them with plasmids with either normal EOS or EOS with a mutation in that protein-protein C terminus binding domain. These cells were also transfected with STAT5 as well. And then we utilized co-IP, which allows us to fish for a protein, essentially, and then determine if the other protein is attached as well. And this is the results of that assay. Looking here, we see our positive controls in our input, and we do indeed see our EOS here and our mutated EOS here as well. And then in our down here, we can see that STAT5 is present in both of our samples. Over here, we see our negative control, and in the middle is the results of this assay. Here we can see that EOS is present once we've pulled down for STAT5. And then once we pull down with STAT5 and our mutated EOS, we are no longer able to see EOS. So this tells us that, one, EOS and STAT5 are able to form a complex, and two, that this complex is likely happening in that C terminus region, because once it is no longer in its natural state, that complex is no longer able to be formed. To further determine where this possible formation or this interaction could be happening, or the role of EOS in Th2 cells, we decided to look at the phosphorylation of STAT5. Remember, STAT5 and other STAT factors need to be phosphorylated in order to be activated and enter into the nucleus. So here there's a time course study at 48, 72, and 96 hours where we look at normal wild-type mice and EOS knockout mice. And we can see that the content of STAT5 is equivalent amongst all of them, but throughout our study with our EOS knockout mice, we have less phosphorylated STAT5, showing that EOS is necessary either for the phosphorylation of STAT5 or possibly the maintenance of STAT5. Taking all this together, we're able to show that there's a novel role for the IKZF family member EOS in the differentiation of Th2 cells. We're able to show that the lack of EOS expression leads to decreased gene and protein expression of multiple gene entities in the Th2 development, as we saw with things such as IL-4, IL-13, PRDM1, and its protein product, BLIMP1. We also saw that the regulation of the EOS and the Th2 gene network occurs through EOS directly binding to DNA, or possibly through a cooperative relationship with STAT5 and its phosphorylation. Understanding these mechanisms are important because the further we're able to delineate how these Th2 cells are able to come about, the more directed our therapeutics can be for autoimmune disorders and any sort of disorder where we have an imbalance of Th2 cells, or in scenarios where we may want more Th2 cells. Again, I would like to thank the Dr. Ken Astrotech Lab, along with its members, Barbara Streetmore, Caitlin Reed, Mike Powell, and then my co-lab mates, Sol Moon and Nikki Sandhu, along with our past members. I'd like to thank FBRI as well, Virginia Tech Carilion, Dr. LeConte, and Dr. Vaughn as well, and all of you for being here today. Thank you, and I'll take any questions.
Great talk. Thank you, Mustafa. Any questions in the room? We're waiting for YouTube questions to start coming in. And while we do, what would be some next steps to take these results towards a clinical?
So there are a lot of studies, I think, think that are in between where we are now and this being a clinical application. I still think there are more steps that can be done to understand the exact role of EOS. Our Th1 data has actually shown that EOS is dependent on the IL-2 cytokine and other cytokines. So one thing that we could do is determine to see if there are other ways that EOS can be regulated in the cell. And then we can also start introducing medications that are currently out there. So an example would be lenalidomide or phthalimide, which actually targets one of the IKZF family members, to see what the downstream effects of that are. So I think we still have some room in terms of understanding the exact role of EOS and the other family members before we can take those steps.
Great. Let's see. We don't have a lot of questions coming in. We'll give them a few more minutes. Okay, well, I think we'll let you go. Thank you for a great talk.
Thank you.
Okay, our last presenter today is going to be Jean Sabile. He worked with Dr. Sami Lamoui at FBRI.
Alrighty, thank you all for having me here this afternoon. So again, my name is Jean Sabile. I work with Dr. Sami Lamoui and Dr. Jennifer Vaughn, and the title of my project is Connexin 43 expression and cytoplasmic localization correlate with colon cancer progression. In terms of what we know about cancer, it's currently the second highest cause of death in the United States, with colon cancer being the third highest in incidence and mortality across all cancers. And when we imagine 100 patients with colon cancer, what we know is that for those with stage 1 and low risk stage 2 disease, up to 90% of these patients treated with surgical resection will survive up to 90 years, up to 5 years, pardon. And high-risk stage 2 and stage 3 disease, up to 71% of these patients treated with a combination of surgical resection and chemotherapy will survive up to 5 years. And unfortunately, for patients with stage 4 metastatic disease, only up to 14% of these patients will survive up to 5 years with chemotherapy. And this is thought to be owed to the widespread nature and the chemoresistant nature of metastatic disease. And so we need to find new molecular targets and therapeutic strategies for colon cancer. And so one of the molecular targets we're studying in the Lamoui lab is Connexin 43. In terms of what we know about Connexin 43 in cancer, favorable prognosis is conferred with increased Connexin 43 in prostate, breast, and non-small cell lung cancer, whereas poor prognosis is conferred with increased Connexin 43 in esophageal, urothelial, and squamous cell carcinoma of the lung. In addition, in the Lamoui lab, prior studies have shown that Connexin 43 interactions with microtubules, specifically in glioblastoma, confer tumorigenic properties. Glioblastoma is a highly aggressive form of brain cancer. And so what do we know about Connexin 43 in colon cancer specifically? It's been shown to be a tumor suppressor, whereas loss of Connexin 43 has been associated with poor prognosis, and an oncogene, where increased Connexin 43 has been associated with late stage and poor outcome. Additionally, Connexin 43 localization shifts from the membrane to the cytoplasm during colon cancer carcinogenesis. And overall, Connexin 43's role in colon cancer has remained unclear. Therefore, it's important to further investigate what is happening with Connexin 43 at an expression level and where it's localized within colon cancer. So in terms of what we know about Connexin 43 itself, we know that it's a 43 kilodalton protein with a known role in cell-to-cell communication, with channel-dependent properties via the formation of gap junctions and hemichannels. And we also know that it has an intracellular C-terminus domain that is known to interact with a variety of intracellular targets that have influence on cellular processes important for cancer, such as cellular proliferation, migration, and apoptosis, so channel-independent roles. And the way we know these interact intracellularly is by direct interaction with the C-terminus with cell cycle regulatory proteins, as well as interaction with protein complexes, with one example being ZO1, a characterized protein that Dr. Gordy's lab at FBRI has also worked on. So in essence, the channel-dependent independent functions account for this dual action as both a tumor suppressor and an oncogene. So it is context dependent. And so from all this, my research has two overall questions and therefore two separate parts. The first part being, what is the expression and localization of connexin 43 during colon cancer progression, and can we target connexin 43 in colon cancer? And the way we began this is with 15 patients that we consented and enrolled in our study with collaboration with Carilion Clinic. And I collected tumor samples with the assistance of Dr. Farrell Adkins at Carilion Clinic, and we separated our patients between early and late stage. Early being defined as stages 1 through 2, and late being defined as stages 3 and 4. And from our tumor samples, we were able to retrieve tumor homogenates, extract protein, and what you're seeing here is a Western blot of connexin 43 across our 15 subjects compared to total protein, which we accomplished via Ponsel red staining. And from this, you can compare connexin 43 to total protein and quantify it to get connexin 43 expression across our 15 subjects. And we conducted odds ratio with connexin 43 elevation as a given exposure, which demonstrated a 4-to-1 probability that given elevated connexin 43, these patients would have advanced stage. In addition to this, we found that when we did t-testing, comparing early and late stage disease with connexin 43 expression, significantly elevated connexin 43 expression in late stage disease. However, we cannot clarify connexin 43's role based on expression levels alone. We have to know where connexin 43 is localized within our primary tissue. And so, before doing this, assessing primary tissues, we must first understand the tissue histology for the various stages of colon cancer. So these are representative images provided to us by Dr. Douglas Grider, our Carilion pathology associate, of the 4 stages of colon cancer across our study subjects. And so overall, what you see is increasing number of malignant glands with increasing variety of size, along with the appearance of negative prognostic indicators such as tumor necrosis, as you see indicated by the black arrows. And so now that we've seen the H&E or hematoxylin eosin staining features of the various stages of colon cancer, we have to look closer at where connexin 43 is actually localized in these tissues. And so we conducted confocal immunofluorescence microscopy across our different subjects, across different stages, beginning first with stage 1, defined at most invasion into the muscularis propria, and here with subject 7. And what you're seeing in seeing in this image is connexin 43 in green, DAPI, which is a nuclear stain, in blue, and CDX2, which is a marker of mature colonic epithelium, in red. And what we found for our stage 1 patients is connexin 43 localized primarily within the stroma, so the fibroblastic element of the disease. And we move on to stage 2, being defined as tumor invasion beyond the muscularis propria, but without nodal or extranodal spread. What we found again is worsening dysplasia of our colonic malignant glands, and again, connexin 43 expression localized within the stroma. And what you're seeing here are connexin 43 plaques, which is representative of gap junction formation. Now, as we move on to stage 3, being again defined by varying tumor invasion into local colonic tissues with nodal spread but without metastases, what we find again is worsening colonic gland disruption with lobulation of these malignant glands, but for the first time only seen in advanced stage are the presence of connexin 43 positive cytoplasmic connexin 43 positive clusters of cells within advanced stage disease. And so these are actually the first images of this phenomenon being taken in colon cancer. This was also seen in metastatic disease, stage 4, with subject 15, and we proceeded to determine whether or not connexin 43 was actually localized within the cytoplasm and took 100x magnification images of these cells and found that connexin 43 was in fact in the cytoplasm of these cells. So what can we conclude from this? We can conclude that increased connexin 43 expression is associated with advanced stage, that clusters of cells that highly express cytoplasmic connexin 43 are observed in advanced stage, and in the future we'd like to characterize the identity of these connexin 43 positive cells and determine whether or not connexin 43 expression is increased at the RNA levels. What we saw was only in protein. So going back to our 2 questions, I showed you that connexin 43 is increased in the cytoplasm of a subset of cells in patients with advanced stage colon cancer, which is exciting, but given the poor outcome in metastatic disease and the fact that tumors are very heterogeneous populations of different cells, we wanted to study connexin 43 in specific cells that are important for tumor metastasis. So in essence, can we target connexin 43 in colon cancer? And so as we know, tumors encompass a subpopulation of cancer stem cells that are important for tumorigenesis. And furthermore, prior studies in other cancers, particularly in glioblastoma, have demonstrated that cancer stem cells may be higher in connexin 43 than non-cancer stem cells. And the way we know cancer stem cells to be tumorigenic in cancer is via the stem cell model, which posits that within a given heterogeneous population of cells, there are populations of highly proliferative cells which we can target biochemotherapy, but often leave behind the more slow-growing quiescent cancer stem cells, which are then free to asymmetrically divide and recapitulate the original tumor. The cancer stem cells also undergo a process called the epithelial mesenchymal transition, which allows them to travel to distal sites where they can then repeat this process and form tumor metastases. And so theoretically, if we have a way of targeting cancer stem cells, we could be vulnerable to populations of highly proliferative cells that are susceptible to the kinds of chemotherapies that we have available. And so we wanted to explore connexin 43 as a target for colon cancer stem cells. So in addition to primary tissue homogenates, we also collected primary cells from our tumor samples of our 15 patients. And so what you see here are representative images of some of our patients and demonstrate how cancer stem cells form tumor spheres in vitro. So we isolated cancer stem cells from non-cancer stem cells and demonstrated here with patient 11 that when comparing cancer stem cells to non-cancer stem cells in colon cancer, that cancer stem cells had increased connexin 43 and with appropriate increases of cancer stem cell markers, CD133, Notch1, and OCT4. And so we reconfirmed our investigations in cancer cell lines, colon cancer cell lines, HCT116 and HT29, and found similarly increased connexin 43 in HCT116 cancer stem cells, whereas we did not see them in HT29. And so to confirm that the increase in connexin 43 seen in HCT116 cancer stem cells was dependent on the cancer stem cell phenotype, we differentiated our cancer stem cells with 10% serum and found a corresponding decrease in connexin 43 upon differentiation of these cells. Now, once more, we can't rely on connexin 43 expression alone to assess connexin 43's role in colon cancer. We must assess its localization. And so we used HCT116 cells and utilized the technique of stochastic optical reconstruction microscopy, or STORM, and found in aqua connexin 43 was very closely associated and localized with microtubules in red. And this is important since this was a similar observation seen in glioma stem cells. And what accounts for this potential interaction is a known tubulin binding domain on the C-terminus of connexin 43. And to study how this interaction between connexin 43 and microtubules might be important for cell survival, we used a mimetic peptide developed by the Gordy lab. Mimetic peptides are named as such since they mime specific regions along the C-terminus of connexin 43. In this case, our peptide GEM2 mimes the tubulin binding domain of the C-terminus of connexin 43. Now, our construct also contains an antennapedia sequence, which allows the uptake of the peptide into the cell, and a biotin tag, which allows visualization of the peptide. In addition, we can generate control peptides, such as here, GEM2 scrambled, which has a scrambled amino acid sequence which does not correspond to the tubulin binding domain. And so to explore if this connexin 43 microtubule interaction could be modulated in a similar way as been shown previously in glioma stem cells, we first assessed whether or not the peptide could be effectively taken up by our colon cancer stem cells. And so again, we conducted confocal immunofluorescence microscopy with our two cancer stem cells from our two cell lines, HT29 and HCT116. With blue, again, DAPI, red is wheat germ agglutinin, which is a membrane stain, and GEM2 being the peptide, and found effective uptake of the peptide in the cells. And then proceeded with treating cancer stem cells from our two cell lines with varying concentrations of GEM2 and our control. And what we found, firstly, with HT29, which again was the population of cells that did not express high levels of connexin 43, the cancer stem cells, no significant change in the formation of tumor spheres in this population of cells. However, when we compared this to the treatment of HCT116 cancer stem cells, again, those expressing high levels of connexin 43 was a qualitatively significant difference in the formation of spheres, which is associated quantitatively with a statistically significant decrease in tumor sphere formation for the HCT116 cancer stem cells treated with GEM2 at 100 micromolars. And so, in conclusion, what we found was increased connexin 43 expression is observed in a subset of colon cancer stem cells, that intracellular connexin 43 co-localizes with microtubules, disruption of the intracellular connexin 43 microtubule interactions via the GEM2 peptide results in decreased colon cancer stem cell survival. And in the future, we'd like to further investigate this potential interaction between connexin 43 and microtubules in primary colon cancer stem cells and test the GEM2 peptide in primary colon cancer stem cells as well. And so with that, I'd like to thank FBRI, Carilion Clinic, VTCOSOM, my peers in the Lemui Lab, the Smith Lab, Sheng Lab, Gordy Lab, again, all our Carilion Surgical Oncology and Pathology Associates, statistics support with Dr. Allison Teggy, all our VTCOSM research curriculum faculty, with specific funding acknowledgement to the American Society of Colon and Rectal Surgeons for the Medical Student Research Initiation Grant I received in spring in 2018. And finally, last but not least, the 15 subjects who donated their tissues and participated in the study, which without participation, none of this would be possible. So with that, I'd just like to thank you all for your time and open it up for questions.
Great, thank you so much, Jean. So I've already got one question here. Ashley Nitre, how did you maintain your cell lines and how do you estimate this compares to in vivo conditions?
That's a good question. So in terms of how we maintained our cell lines, you have to be specific of what specific cell lines we're talking about. In terms of adherent, quote unquote, normal cancer cells, we used RPMI medium with 10% fetal bovine serum. So the critical thing is the serum allows the cells to maintain their adherent differentiated state. Without serum, you can isolate the cancer stem cells in that population, as the adherent cells will die without the serum. So that's how we maintain those two populations of cells. And the second question was?
Difference between in vivo.
Yeah, so obviously the microenvironment in vivo is significantly different than the controlled environment within a flask. And so there's many, many different interactions between not just cancer cells, but also the supporting fibroblasts that you saw on the confocal imaging that could have a role to play in mediating a lot of those interactions.
Great, we've got another question. Dr. Jamie Smith asks, how do you think other connexins, for example, connexin 32, connexin 26, might be involved here? Do you think they would act independent to or cooperatively with connexin 43's cytoplasmic role?
Yeah, so different connexins have been shown to have a role across different cancers as well. So 46 and 32, I know, have been shown to be active, I believe, in breast cancer as well. But in terms of the general concept of the different connexins essentially interacting, we know that heteromeric channels are possible. So in terms of the channel functions, heteromeric channels, so a combination of connexin 43, 32, 26, may have yet unknown roles to play in tumorigenicity. And again, it varies by cancer, whereas connexin 43 elevation confers poor prognosis and favorable prognosis depending on the cancer. So it may be, those channels may act differently across different cancers as well.
Another question here from Miranda Gerard. Awesome presentation. Has anyone looked to see if connexin 43 is unregulated in younger patient populations with colorectal cancer as well, given the rising incidence in this age group?
Yeah, so you point out the bimodal distribution of colon cancer in younger patients, specifically like 40. At this point, I don't believe that anyone's looked at connexin 43 specifically as something that would be unregulated in this specific population of patients. I think the data collection for this younger patient population is still being collected, but it's an interesting question. Now that we have a tissue repository, it could be a future work.
That's a good point. I'm going to do one more question from online. David O'Neill, nice work. Given the widespread presence of connexin 43 in various healthy tissues, is there any evidence that cancerous connexin 43 is mutated or unique to mitigate toxicity of a targeted therapy? Sure. So, given connexin 43's presence in healthy tissue, is there any evidence that connexin 43 in cancerous tissue is mutated or somehow unique in order to, worried about the toxicity to healthy tissues if you're going to target it?
It's an interesting point. Obviously, connexin 43 is really important parts to play in myocardial contractility and whatnot. I'm not sure if we have evidence to say in colon cancer specifically if there's any mutated connexin 43 as a role to play. We do know that there are different phosphorylation states of connexin 43 that have varying roles, as well as smaller kilodalton isoforms of connexin 43, and it's unclear if those have a role in disease states compared to normal. So it's a good question.
Great. Okay, well, we are going to end with our questions now and move on to the awards portion. Thank you so much, Jean, for your presentation and answering your questions. So I'm going to wrap up our symposium today, but before I do, we have a few more awards I want to go ahead and let you know have been awarded, as well as a message from one of our beloved faculty members for the class of 2020. She'll be zooming in here in just a minute. But first I want to go ahead and read off the names of the winners of the Letters of Distinction in Productivity. So the Letters of Distinction in Research Productivity were selected based on students' overall scholarly activity, with an emphasis placed on publications and presentations of the formal research domain project, as well as any grants that may have come from that. And the scholarly activity we considered including publications, oral and poster presentations at regional and national meetings, awards, grants, and we also take into consideration research work that our students have done outside of their formal research domain projects. So many of our students have been engaged in a number of different ways throughout their time at VTCSOM. And so I'm going to read off the class of 2020 winners. They are the ones who will eventually be receiving their red ribbons that are up on the table behind me. And so class of 2020 research letters of distinction in productivity. First one is Malek Bouzaher, worked with Dr. John Chappell at FBRI. The title of Malek's project was Parasite Coverage and Microvessel Caliber in Normal and Preeclamptic Pregnancies. So we heard a little bit of that study when Anisha Chadha presented earlier. Malek had 2 poster presentations of his research domain project, as well as 8 publications and additional presentations of work outside of his research domain project. The second letter of distinction in productivity winner was Brendan Fusco. His mentor was Dr. Alexei Morozov at FBRI. The title of his project was Artificial Potentiation of the Dorsomedial Prefrontal Cortex to Basolateral Amygdala Pathway. His project was actually published recently in Neurophotonics, and he also gave a poster presentation at the Society for Neuroscience. Another letter of distinction winner in productivity was Tom Liu, who we heard from earlier today, worked with Rob Gordy. He was a recipient of the American Heart Association Scholarship in Cardiovascular Disease. He did posters at 3 different conferences on that work, and then he had several publications and presentations on research outside of research domain. Bobby Nicholson is also a recipient of the Letter of Distinction in Productivity. His mentor is Dr. Damon Keel. Lost my place here. Dr. Damon Keel was his mentor in the Department of Emergency Medicine. The title of Bobby's project is Utilization of Referral to a Prescription Exercise Program to Reduce Pain and Improve Function Following Discharge from the Emergency Department. Bobby had an oral presentation at the Society for Academic Emergency Medicine regional meeting, 2 poster presentations, and is in the process of, is that mine? Sorry, lots of feedback here. Okay, Brian rescues us again. And his manuscript is currently in preparation for submission to the Journal of Physical Activity and Health. Hannah Palmerton worked with Dr. Gary Simmons in the Department of Surgery, and she worked on a project entitled The Effect of Various Operating Room Noise on Fine Motor Skills, Cognition, and Mood. So she published her work in the Journal of Surgery Research and Practice and had a poster presentation at the Congress of Neurosurgeons annual meeting. Brian Sawe also earned a Letter of Distinction in Productivity. He worked with Dr. Terry Ann Watzman and Lori Seidel, Departments of Surgery and Psychiatry. The title of his project is Mindful illness in the OR. He had 2 publications related to his research domain project published, and acceptance to present a poster at the American Association of Neurological Surgeons, as well as several publications and presentations outside of research domain. Farah Shah worked with Dr. Z. Xing at FBRI. The title of her project was PIK3CB and connexin 43 inhibition sensitizes glioblastoma cells to temozolomide. Farah was recipient of several summer research fellowships to support her research, including the AOA Carolyn L. Kukine Research Fellowship, St. Baldrick's Foundation Fellowship, an American Academy of Neurology Summer Research Scholarship. She also had 2 poster presentations, as well as a publication and presentation outside of research domain. And finally, the last in my list, recipient of Letter of Distinction and Productivity is Ben Walker. He worked with Mark Hamill in the Department of Surgery, and the title of his project was The Effects of Interdisciplinary Education Regarding Potentially Inappropriate Medication Use in Geriatric Fall Patients. He actually published his work in 2 first author publications in the American Surgeon, as well as another publication in the Journal of Trauma and Acute Care Surgery, and had an oral and poster presentation at the Southeastern Surgical Congress. So in sum total, the class of 2020 has been very productive, and I know many of our students are in the process of preparing publications for submission. So we're looking forward to continuing to see that productivity grow over the course of the next year or 2. So I think now we are prepared to take a moment to switch over to hear from Dr. Jen Vaughn. So Dr. Jen Vaughn worked with the class of 2020 for several years and recently left VTCSOM, but she would like to say a few words to you.
Starting over, hello to the class of 2020 from Ohio. It had always been my plan to be there with you all today to support you on this exciting day, but unfortunately, these trying circumstances have been challenging for us all. I wanted to wish you all congratulations on a fantastic match. I'm so excited to see the results.
Dr. Vaughn, if you're listening to yourself on YouTube, please speak up.
And I wish you all the best in your endeavors going forward to your residencies as you enter the front lines in the next few months. I'm always here if I can be of any assistance to any of you. It's been an honor to work with you these last 4 years and see you develop as scientist physicians. Take care of yourselves and each other.
Thank you so much. Thanks for joining us. Okay, can we switch back here? Okay.
Okay, thanks everybody for being patient with us as we work through our technology challenges here. I'm gonna say a few words of thanks before we get to what I know everybody's been waiting for, which is the results of the votes on the posters. I want to say thank you to so many people. Dr. Tegi has helped so many people with their research projects for her research support and guidance. Dustin, Ryan, who've all been working overtime to get this working, jumping through all these hoops when I asked them. Yes, applauding. We are applauding. Thank you so much. This has been very difficult to pull off, and I really appreciate everybody jumping in and making this happen. Andre Dean as well. Van Greek helped out with the website, which I think is a challenge to adapt as we had to figure out what we were going to be doing. Allison Matheson, Catherine Doss helping everything get up and running, and Crystal Barnett and Jessica Flowers. Jessica, who is also at home watching, helping be our eyes and ears at home. Thanks so much for putting up with all of these changes. Thank you, Crystal and Jessica. I think the day went very well. Thanks to everybody who attended and commented on YouTube. I really appreciate it. And I can't forget Dr. Brock Mutchison, who has been scrambling to pull together our Qualtrics survey results, which he has done. So we are, I think, going to switch to a slide that's going to give us the results of that. Is that correct? But I can read them here. Okay. So, we had several awards. The first award was Most Tweetable Results. And so the Most Tweetable Results award goes to Mercedes Robinson, poster number 31. The Best Visual Design poster award goes to Maggie Neifel, poster number 15. The Best Poster Presentation, as viewed on our 5-minute poster presentations, goes to Hirsch Patolia, poster number 28. The Most Hurdles Overcome, I know there were many of you, and I wish I could have voted for many of you myself, but the Most Hurdles Overcome goes to Judy Chen, poster number 3. Most Exciting Discovery, and I think there were several of those where I would have voted as well, but this award goes to Chris Liao, with poster number 16. Then we had Most Likely to Change Clinical Practice this year, and that award goes to Brian Sawe. And then finally, Most Promising Preliminary Data for an NIH grant goes to Jean Sabile, who we just heard from. So congratulations to everybody who won those awards. I will be sending out a virtual award to you. It's not imaginary, it's virtual, so you'll actually be getting something in your email. The same goes for those of you who offered several excellent questions on the YouTube stream. Thank you all for your participation, and thanks for your support today. All right, that's it.